Hello and welcome to the Drift to the Stars. Wherever you are tonight, I'm glad you found your way here. This is your moment to slow down. Let the noise of the day grow distant. Let your thoughts settle, like dust drifting quietly to the floor of a still room. Tonight, we're staying close to home, right here on the planet we've always called our own. Earth is stranger and more beautiful than most of us ever take the time to notice. From glowing seas to forests that think, from creatures that defy death to the hidden light your own body quietly emits, this world is alive in ways that will gently surprise you, soothe you, and stir something tender and deep. Some of what you hear tonight may shift the way you see the world. Other things may simply drift past like shadows on water, soft, curious, and quietly unforgettable. If this kind of slow wonder brings you peace, consider liking and subscribing. It helps others find their way here too, one sleepy soul at a time. But for now, make yourself comfortable. Take a long, slow breath, and let's begin this gentle journey through the most extraordinary place we know. In the warm coastal waters of the Mediterranean and in oceans scattered across the globe, there drifts a creature that has quietly broken one of nature's most fundamental rules. It is called Turritopsis dohrani. Most know it simply as the immortal jellyfish. When this small, translucent animal is damaged, threatened, or simply worn by age, it does something no other creature on Earth does quite so completely. It turns back. Like a butterfly reversing its own transformation and returning to the caterpillar it once was, the jellyfish reverts to its earliest form, a tiny polyp, the biological equivalent of childhood, and begins its life cycle again from the start. This is not legend or metaphor. It has been observed repeatedly in laboratory conditions. The jellyfish cycles through its life stages again and again, potentially without limit unless something in the world around it intervenes. It holds no secret formula for human immortality. It offers no shortcut through time. But it does offer something rarer, a reminder that the rules we take for granted are only rules until nature decides otherwise. Here, drifting in silence beneath the surface of the sea, is a creature that rewrites the ending. Not once, not twice, but endlessly in the dark and quiet water, beginning again. In the stillness of a forest, something is happening beneath your feet that no amount of quiet walking will ever reveal. Trees, long imagined as solitary and self-contained, are in constant communication. Threading through the soil beneath them is a vast web of fungal filaments known as mycorrhizae, delicate, hair-thin connections that link root to root, tree to tree, across entire forests. Through this hidden network, trees exchange nutrients, pass chemical warnings about approaching insects or spreading drought, and share resources with neighbors in need. Older trees, sometimes called mother trees, have been shown to funnel sugars and minerals down through this web to support younger, struggling seedlings growing in their shade. Scientists have taken to calling it the Wood Wide Web. And the name is not entirely playful. The network operates with a complexity and reach that genuinely mirrors the systems we once thought only humans could build. A birch may sustain a neighboring fir. A dying tree may spend its final energy sending everything it has left outward into the roots of those around it. It is not conscious. It does not think or feel in any way we would recognize. But it cooperates, quietly, persistently, and across distances that would take us hours to walk. Beneath the forest floor, the trees are never truly alone. They are less than a millimeter long. They have 8 stubby legs, a wrinkled body, and a face that looks, under a microscope, like something between a bear cub and a tiny alien. They are called tardigrades, though most people who encounter them prefer their nickname, water bears. Do not let their size or their charm mislead you. Tardigrades are among the most resilient organisms ever discovered. They can survive temperatures close to absolute zero and heat well beyond boiling. They can endure radiation levels that would be lethal thousands of times over to any other known animal. They can withstand pressure 6 times greater than the deepest ocean trench. They can live without water for decades. And in 2007, scientists placed them aboard a spacecraft and launched them into open orbit, into the vacuum of space, into unfiltered solar radiation and the full cold silence of the cosmos. They came back alive. Their secret is a process called cryptobiosis. When conditions grow unbearable, a tardigrade draws nearly all the water from its body, folds itself into a tiny dormant capsule called a ton, and suspends its metabolism almost entirely. It does not die. It simply waits, sometimes for years, sometimes for decades. And when water returns, it wakes. In the quiet corners of moss and soil and melting ice, the water bear waits. Patient, ancient, and almost impossible to end. Every breath you take tonight is the legacy of a catastrophe. More than 2 billion years ago, Earth's atmosphere contained almost no oxygen. The planet's earliest inhabitants, microscopic organisms called anaerobes, had no need for it. They thrived in its absence, drawing energy from the ancient chemistry of a young and airless world. Then came the cyanobacteria. These tiny photosynthetic microbes began releasing oxygen as a byproduct of their activity. At first, almost nothing changed. The oceans absorbed it. The rocks absorbed it. But slowly, over millions of years, oxygen accumulated in the atmosphere, and the world began to shift. For the anaerobes that had ruled the planet, this was not a gift. It was a poison. Oxygen corroded their cells, disrupted their ancient chemistry, and drove the majority of them to extinction in an event scientists now call the Great Oxidation Event, one of the most sweeping mass extinctions in Earth's entire history. But for other organisms, the ones that could tolerate or even harness this strange reactive gas, it became something extraordinary, a new and powerful source of energy, the foundation upon which all complex life, every animal, every plant, every creature that has ever drawn a breath, would eventually be built. The air that keeps you alive tonight was once the world's most dangerous pollutant. Every inhale carries that history quietly within it. We tend to imagine plants as passive things, patient, gentle, rooted in place, reaching softly toward the light. But not all of them wait. Carnivorous plants have evolved in some of the most nutrient-poor places on Earth. Boggy soils and rocky outcroppings where nitrogen is vanishingly scarce. Unable to draw what they need from the ground, they turn to another source entirely. They began to hunt. The Venus flytrap lures insects with the promise of nectar, then monitors them with extraordinary precision. It counts. When trigger hairs inside its open jaws are touched, not once, but twice, or the same hair twice in quick succession, the trap snaps shut in a fraction of a second. A single touch is not enough. The plant is too careful for false alarms. Pitcher plants form deep, elegant wells lined with slick walls and filled with digestive fluid. Insects are drawn in by scent, lose their footing, and cannot climb back out. Sundews extend glistening tentacles coated in sticky droplets that look to a passing insect like dew. The digestion of the insect, extracting the nitrogen and other nutrients the soil cannot provide, takes between 5 and 12 days. Then the trap reopens dry and empty and waits again. No nervous system coordinates this. No brain decides. A plant is measuring time and counting touches and acting on what it finds quietly and precisely in the Carolina sun, on the rocky, windswept shores of Antarctica, where the landscape offers almost nothing soft and the cold is a constant and the ground is frozen beneath the feet. Penguins build their nests from the only material available in abundance: stone. For Adelie and Gentoo penguins, the nest is a shallow cup of carefully selected pebbles, raised slightly above the surrounding ground to keep eggs clear of snowmelt when temperatures rise. A well-constructed nest, with good drainage and sufficient depth, can mean the difference between a clutch of eggs that survives and one that does not. The quality of the pebbles matters. Their selection is not casual. When something lands, the tentacles curl inward with slow, patient deliberation. When it is time to form a pair bond, a male Adelie penguin searches the available ground for the smoothest, most symmetrical stone he can find. He carries it to the female of his choosing and presents it to her, lowering his head in a small, deliberate bow. If she accepts the pebble, if she takes it and places it in the hollow she has begun to scrape in the earth, the pair is formed. They will build their nest together, stone by stone, over the days that follow. Pebbles are among the most valuable commodities in a penguin colony. They are stolen from neighboring nests when the owners are not watching, defended with considerable energy, and sometimes exchanged between individuals in transactions that researchers have described with some caution as resembling barter. But the first pebble, the one offered and accepted, is something else. A beginning. A small, cold, quietly serious act of devotion in one of the most inhospitable places on Earth. Above the open ocean, far beyond the sight of any coastline, a frigate bird soars on thermals that carry it thousands of meters above the surface of the sea. No teeth. It has been airborne for days. It may remain airborne for weeks. There is no land to rest on, no perch to settle upon, no surface below it except water that it cannot land on without difficulty. And yet, at some point, it must sleep. Researchers tracking frigate birds with instruments capable of monitoring brain activity discovered something that resolved what had long been an open question in biology. While soaring on updrafts, frigate birds do sleep, but they do so with only one hemisphere of the brain at a time, a phenomenon called unihemispheric slow-wave sleep. One side rests while the other remains active, monitoring the bird's position, maintaining the adjustments of flight, watching for obstacles or changes in wind. The eye connected to the sleeping hemisphere closes. The eye connected to the waking hemisphere remains open. Then the hemispheres switch. No claws. Full sleep, in which both hemispheres rest simultaneously, does occur during flight, but only in very brief episodes, typically lasting seconds. The bird appears to reserve deeper rest for those moments when it is riding a stable thermal with minimal need for active correction. On land, frigate birds sleep normally, with both hemispheres resting together. But when the ocean stretches endlessly beneath them and there is nowhere to descend, they have found a way to carry rest with them into the sky. Even at altitude, in the dark, on the wind, something in them is always watching. Right now, even as you lie still and your eyes grow heavy and the world outside softens into background, something extraordinary is happening inside you. Your brain, a structure of roughly 4 kilograms of soft tissue, the consistency of firm gelatin, suspended in fluid and enclosed in bone, is producing thought. Not metaphorically, physically. Through electricity. No pursuit. Each of the approximately 86 billion neurons in your brain is a living cell capable of generating and transmitting electrical signals. When a neuron fires, an electrical impulse travels along its length, driven not by wires or circuits, but by the movement of charged ions through protein channels embedded in the cell membrane, and reaches the junction where it meets the next neuron. There, it triggers the release of chemical messengers called neurotransmitters that cross the microscopic gap between cells and initiate, or inhibit, the firing of the next neuron in the chain. From the coordinated firing of billions of cells in patterns that shift moment to moment, arise sensation, memory, language, imagination, and the quiet experience of being a self in the world. The patterns are never the same twice. Every thought you think, every image you hold in your mind, every feeling that passes through you corresponds to a unique configuration of electrical activity distributed across the tissue of your brain, arising and dissolving in fractions of a second. Even now, as wakefulness recedes, the patterns change. The brain does not go dark in sleep. It shifts. It consolidates. Just stillness, design, and a quiet hidden appetite. It processes. The storm continues, quieter now, carrying you gently under. In the shallow coastal waters of the Indo-Pacific, resting on a patch of sand or tucked against a coral formation, a cuttlefish attends to the business of being invisible. Within a fraction of a second, its skin transforms. Pigment cells called chromatophores, each one a tiny elastic sac filled with colored pigment, controlled by muscles connected directly to the nervous system, expand or contract across the skin surface in coordinated patterns, producing colors and shapes with a precision that no other animal in the ocean can match. Beneath them, iridophores reflect and manipulate light at the structural level, adding iridescence and depth. Papillae, small muscular projections embedded in the skin, rise or flatten to alter the surface texture, matching the roughness of coral, the smoothness of sand, or the irregular surface of encrusted rock. The result is camouflage of a quality that frequently makes the cuttlefish functionally invisible to both predators and prey. A cuttlefish can settle onto a pebbled seafloor and within moments become indistinguishable from it. What makes this more remarkable still is that cuttlefish are colorblind. In the most difficult places on Earth, where survival demands creativity, even the most delicate green leaf can become a trap. Their eyes contain only a single type of photoreceptor, meaning they perceive no color in the conventional sense. How they manage to match the colors of their surroundings so accurately without being able to see those colors is a question that researchers are still working to answer. Current evidence suggests they may use the polarization of light or the brightness and contrast of patterns as proxies for color information. The cuttlefish disappears. The ocean continues around it, apparently empty, knowing nothing of what it contains. On a specific night each year, or sometimes just a few nights, occurring within a narrow window dictated by water temperature, day length, and the phase of the moon, something happens on coral reefs across the tropical ocean that has no real equivalent anywhere else in the natural world. The coral spawns. All at once, across reefs that may extend for hundreds of kilometers, individual coral colonies release tiny bundles of eggs and sperm simultaneously into the water column. The bundles rise toward the surface in a slow, dense upwelling, a blizzard of life ascending through the dark water, pink and white and pale orange in the beam of a diver's light. The synchrony is remarkable. Hold a leaf up to the light and for a moment let yourself really look at it. Corals are sessile animals. They cannot move, cannot seek out partners, cannot coordinate their behavior through any direct communication. And yet colonies separated by kilometers release their gametes within the same narrow window of hours, sometimes within the same minutes. The cue appears to be a combination of factors: water temperature reaching a seasonal threshold, the lengthening of days beyond a certain point, and the light of a full moon providing the final trigger. The lunar cycle's influence is precise enough that spawning on many reefs occurs within a predictable number of nights after a specific full moon each year. In the open water, the gamete bundles break apart as they rise, eggs and sperm mixing in concentrations high enough to make fertilization likely despite the vastness of the ocean around them. The fertilized eggs drift, develop, and eventually settle as new polyps on the reef below. The moon calls. Across hundreds of kilometers of dark water, the reef answers all at once. A frog sits at the edge of a still pond, motionless except for the subtle pulse of its throat. An insect moves within range. Spreading across its surface is a branching network of veins, fine as rivers seen from high above, carrying water and minerals to every corner of the leaf and drawing sugars back toward the stem. The frog's tongue, long, sticky, and attached at the front of the mouth rather than the back, fires outward with extraordinary speed, contacts the insect, and retracts, pulling the prey into the open mouth. And then the frog blinks. Not a social blink, not a reflexive response to light or dryness, but a deliberate and functional one. As the frog's eyelids close, the eyeballs are drawn downward and inward into the skull, pressing against the roof of the mouth from above. This pressure, gentle but consistent, pushes the prey toward the back of the throat, assisting the muscles of swallowing in moving food in a direction that the frog's anatomy makes otherwise difficult. The phenomenon has been observed and measured carefully. High-speed cameras have captured the retraction of the eyeball during swallowing, and studies have confirmed that removing this contribution meaningfully reduces swallowing efficiency. The eyes are not merely incidentally involved. They are doing something. It is a solution that only makes sense in the context of the specific anatomy of a frog, a creature whose eyes are large relative to its skull, positioned high on the head and capable of being retracted significantly into the orbit. The architecture is not random. Evolution, working with what was available, found a use for the eyeball that no engineer designing a swallowing mechanism from scratch would ever have arrived at. In the world of frogs, even the act of seeing is, when required, put to work. A caterpillar settles onto a leaf and begins to eat. From the perspective of the plant, this is not a passive event. Within minutes of tissue damage beginning, chemical changes are already underway in the affected leaves. Signaling compounds are synthesized and transported through the plant's vascular system, triggering defensive responses in undamaged tissues, the production of compounds that make the leaves less digestible or more toxic, or simply less palatable to the insect that is consuming them. But the response does not stop at the boundary of the individual plant. Some plants under attack release volatile organic compounds into the air, specific blends of chemicals that drift outward from the damaged tissue and can be detected by neighboring plants of the same or related species. When neighboring plants detect these airborne signals, they begin mounting their own defenses, producing protective compounds before any insect has reached them. The signals can carry information about the nature of the threat. It is precise, efficient, and unlike any other leaf that has ever existed. A plant being chewed by a caterpillar releases a different chemical blend than one being pierced by an aphid or infected by a fungal pathogen. Neighboring plants respond accordingly, tailoring their defenses to the specific type of damage the signal implies. This chemical conversation is not conscious. The plants have no awareness of what they are doing or why. And yet the system functions with a specificity and a reliability that goes considerably beyond what we once assumed plants were capable of. The forest is not quiet. Between every leaf and its neighbor, in the invisible medium of the air, information is moving. Warnings are being sent. And somewhere nearby, a plant that has not yet been touched is already preparing. Beneath nearly every garden, meadow, forest floor, and agricultural field in the temperate world, they are working. Scattered across the surface are thousands of tiny pores called stomata, valves that open and close to regulate the exchange of gases, letting carbon dioxide in and releasing oxygen out. Blind, voiceless, and moving through the darkness at a pace measured in centimeters per hour, earthworms are among the most consequential animals on Earth. An earthworm feeds by ingesting soil, passing it through its digestive tract, and expelling it as castings, material that has been broken down, chemically altered, and enriched with microbial activity. The castings are significantly more nutrient-rich than the surrounding soil, providing plants with nitrogen, phosphorus, potassium, and other essential elements in forms more readily absorbed by roots. As worms move through the soil, they create burrows. These burrows aerate the soil, creating channels through which water can drain and air can penetrate, preventing the compaction that would otherwise make it difficult for roots to grow and for rainfall to be absorbed rather than running off the surface. In heavy rain, a soil populated by earthworms absorbs water many times faster than one without them. Charles Darwin spent decades studying earthworms, ultimately concluding that few other animals had played so significant a role in shaping the history of the land. He estimated that the earthworm population of a single English acre turned over approximately 18 tons of soil per year. They have been doing this for hundreds of millions of years, long before the first grasses appeared, long before the first flowering plants, working patiently in the dark beneath whatever grows above them, making the ground ready for whatever comes next. Standing up to 6 meters tall and weighing as much as 1,000 kilograms, the giraffe is the tallest living terrestrial animal on Earth. They respond to light, to humidity, to the chemical signals of the plant itself. Its heart is enormous. It has to be, to pump blood up that extraordinary neck to the brain. Its legs are long enough that lying down and standing up are both effortful, slow, and during those transitions, briefly dangerous. Perhaps partly for this reason, giraffes sleep less than almost any other large mammal. In the wild, a giraffe typically rests for a total of between 30 minutes and 2 hours per day, taken in short intervals scattered across the night. For much of this rest, the animal remains standing, reducing the time and effort required to rise quickly if a predator approaches. True deep sleep, during which the giraffe lies down and curls its long neck back along its body in an elegant arc, occurs only in brief episodes, sometimes lasting just a few minutes at a time. A giraffe lying down is a giraffe that cannot stand quickly. In lion country, that vulnerability has real consequences. How a large, metabolically active mammal sustains full cognitive and physical function on such minimal sleep is not entirely understood. Studies of sleep requirements across mammals suggest that sleep duration correlates broadly with body size, larger animals tending to need less, but the giraffe's requirements appear low even within that pattern. They are, in their way, breathing. In the moonlit savanna, surrounded by the sounds of the African night, the giraffe stands and watches, resting only in moments, its great height an advantage and a constraint simultaneously, always half present in the world. Plastic was, for most of the 20th century, considered essentially indestructible on any human timescale. A plastic bottle, buried in soil or dropped in the ocean, would persist for hundreds of years, perhaps thousands, breaking down into ever smaller fragments but never truly disappearing, never being absorbed back into the biological cycles that reclaim almost everything else. Then, in 2016, researchers in Japan announced the discovery of a bacterium living in the sediment outside a plastic recycling facility that was doing something no microbe had previously been confirmed to do. It was eating PET, polyethylene terephthalate, one of the most common plastics in global use, found in water bottles, food packaging, and synthetic fibers. The bacterium, named Ideonella sakaiensis, produced 2 enzymes capable of breaking the chemical bonds in PET, reducing the plastic to simpler compounds it could absorb and metabolize. Since that discovery, other plastic-degrading organisms have been identified. Certain species of fungi have been found capable of breaking down polyurethane. Wax moth larvae and the bacteria living in their guts can degrade polyethylene. Researchers have identified plastic-consuming microbes in environments ranging from deep-sea sediment to the digestive systems of insects to the surface waters of the open ocean. The green that catches your eye is chlorophyll, the pigment that captures the energy of sunlight and transforms it into the sugars that fuel nearly every food chain on Earth. The organisms are not fast. The degradation they perform happens over months or years, not days. But they are real. And they are evolving, adapting in the span of decades rather than millennia to a material that did not exist on Earth until we made it. Life, as it has always done, is finding a way to use what is available, even the things we left behind. In the northernmost reaches of the world, where the landscape alternates between months of white and months of grey-green thaw, the Arctic fox moves across the tundra in a coat that is never quite the same as it was 6 months before. In winter, the Arctic fox is white. Not off-white, not cream, but a dense and luminous white that renders it almost invisible against snow and ice. A camouflage so effective that a fox lying still in a winter landscape can be nearly impossible to detect even at close range. The fur is also, by the measurement of insulating capacity, among the warmest of any mammal studied. Without it, almost nothing you know of as alive would be. Arctic foxes have been recorded remaining active and comfortable at temperatures approaching -70 degrees Celsius, conditions under which most mammals and most survival equipment would fail. As spring arrives and the snow recedes to reveal the grey and brown and muted green of the summer tundra, the white coat is shed. In its place grows a shorter, denser coat of brown or grey, matched now to the rocks and soil and low vegetation of the ice-free ground. This twice-yearly transformation is not merely cosmetic. It is a precisely timed biological process, triggered by changes in day length detected through the eyes, coordinated by hormonal signals, and executed through the replacement of millions of individual hairs across the entire body surface. The Arctic fox does not choose its disguise. It becomes it, shaped by the season as surely as the landscape around it is shaped, the land and the animal changing together as the year turns. Drop below 200 meters in the ocean and sunlight ceases. Not dims, ceases. The last blue wavelengths are absorbed by the water above, and what remains is a darkness more complete than most people living on land ever experience. Some leaves track the movement of the sun across the sky, tilting slowly throughout the day to maximize the light they receive. Below 1,000 meters, the pressure is 100 times that at the surface. The temperature hovers just above freezing. There is no plant life, no photosynthesis, no daily cycle of light and dark to organize existence around. And yet the deep ocean is full of life. The anglerfish carries its own light source, a bioluminescent lure dangling from a modified spine above its enormous mouth, drawing curious prey toward it in the dark. The barrel-eye fish has transparent fluid-filled domes above its head, housing upward-pointing tubular eyes that gather the faintest traces of downwelling light with extraordinary sensitivity. The vampire squid, despite its name, feeds not on blood, but on marine snow. The slow, continuous drift of dead organic matter sinking from the productive surface waters above, which in the deep ocean represents the primary source of incoming energy. Siphonophores drift through the midwater as chains of specialized individuals, technically a single organism, but one that can extend for tens of meters, longer than a blue whale. Comb jellies pulse with iridescent light. Dumbo octopuses move with the unhurried grace of animals that have never needed to be fast. Others repel water so completely that droplets bead and roll off without leaving a trace. We have explored only a small fraction of the deep ocean. What has been seen is already stranger than most imagining. What remains to be discovered is, in all likelihood, stranger still. The most alien world accessible to us is not beyond the atmosphere. It is beneath the waves. For much of the 20th century, the use of tools was considered a defining characteristic of human beings. Then, in 1960, Jane Goodall observed a chimpanzee stripping the leaves from a twig and using it to extract termites from a mound, and the definition required revision. Tool use, it turned out, was not uniquely human. But it is also not uniquely primate. New Caledonian crows manufacture tools with a sophistication that remains remarkable even after decades of study. Some store heat. They select specific plant materials, trim and shape them into hooks and probes, and use these instruments to extract grubs from crevices in wood, a task their beaks alone cannot accomplish. They have been observed making tools from materials they have never previously encountered in the wild, improvising solutions to novel problems in real time. Green herons have been documented dropping small objects, insects, feathers, pieces of bread, onto the surface of water as lures, waiting for fish to approach and then striking. The behavior has been observed in multiple individuals across different populations, suggesting it has been learned and potentially passed on. Woodpecker finches on the Galapagos Islands use cactus spines and small twigs to probe for insects in bark and crevices. Egyptian vultures carry rocks in their beaks and use them to crack open ostrich eggs too large and thick-shelled to be broken by beak strikes alone. In each case, the bird identified a problem, assessed the available materials, selected or modified a tool appropriate to the task, and used it effectively. The capacity to look at the world and see in some object within it a potential solution. That capacity, it appears, has evolved more than once. The camel occupies a specific and demanding ecological niche, the hot desert, and every aspect of its biology reflects the demands of that niche with a thoroughness that amounts to something close to perfection. Begin with the hump. Some produce toxins. It does not store water, as the persistent popular belief holds. It stores fat, a concentrated energy reserve that can sustain the animal through extended periods without food, and that yields some water as a metabolic byproduct when it is broken down. A well-nourished camel's hump stands firm and upright. A camel that has been without food for an extended period carries a hump that has deflated and tilted to one side, the fat reserves depleted. The rest of the camel's water management is no less sophisticated. Its kidneys are among the most efficient of any mammal, producing urine of extraordinary concentration and retaining water that other animals would lose. Its feces are dry. Its body temperature fluctuates by several degrees across the course of the day, absorbing heat during the warmth of daylight and releasing it during the cooler night, a strategy that reduces the need for evaporative cooling through sweating. Its red blood cells are oval rather than circular, a shape that allows them to continue flowing through the bloodstream even when the blood thickens significantly during dehydration. Long eyelashes shield the eyes from blowing sand. Some, as we have already seen, trap and digest insects. A double row of lashes provides additional protection. The nostrils can be closed entirely. The broad, padded feet distribute weight across loose sand without sinking. When water is finally available, a camel can drink more than 100 liters in a matter of minutes, rehydrating with a speed that would be dangerous for most mammals. Every part of the camel is an answer to a question the desert has been asking for millions of years. Inside a hen's egg, in the warm stillness of the nest, days before the shell will crack, a sound is already being made. The developing chick, not yet hatched, still enclosed in the darkness of the egg, dependent on the yolk and the careful regulation of temperature by its mother's body, produces soft, rhythmic vocalizations that pass through the shell and into the air outside. These are not random sounds. They are calls, and they receive responses. The mother hen sitting on the nest calls back. No two are shaped exactly alike. She adjusts her behavior in response to what she hears, shifting her position on the eggs, modifying the sounds she produces, responding to changes in the tempo or character of the chick's calls with changes in her own. The exchange has the character of a conversation conducted through the barrier of the shell between a mother and a chick that has not yet entered the world. Unhatched siblings in adjacent eggs respond to one another as well. In clutches where multiple eggs are developing simultaneously, the calls of one embryo can influence the development and hatching timing of others, helping to synchronize the emergence of the brood so that the hen is not managing hatchlings at dramatically different developmental stages simultaneously. Researchers studying this prenatal communication have found evidence that it influences the development of the chick's auditory system, its stress responses, and its early social behavior, that what passes between mother and embryo through the wall of the egg has consequences that extend into the chick's life long after the shell has been left behind. The egg is not a sealed silence. It is, from early in development, already a place of exchange. Winter arrives in the northern forests with a thoroughness that leaves small creatures very few options. Migrate if migration is possible. Find shelter deep enough that the temperature remains above freezing. Each one is a complete and functioning world, fragile, purposeful, and quietly extraordinary. Or die and leave behind eggs or larvae to carry the lineage forward when warmth returns. Some insects, however, have found a fourth way. They freeze, and they survive it. The goldenrod gall fly overwinters as a larva inside the woody gall it has induced on a goldenrod stem, a structure that offers almost no insulation, exposed entirely to the ambient temperature of the winter air. When that temperature drops below freezing, the larva freezes with it. Its body fluids solidify. Its metabolism effectively ceases. It survives this through the production of cryoprotectants, chemical compounds, principally glycerol and sorbitol, that it synthesizes in large quantities in the weeks before winter arrives. These compounds prevent ice from forming inside its cells, where ice crystals would rupture membranes and destroy the cellular machinery. Imagine standing at the edge of the sea on a dark, quiet night. Ice does form in the spaces between cells, but the cells themselves remain liquid and intact. The woolly bear caterpillar, the larva of the Isabella tiger moth, can spend up to 14 winters in this frozen state, thawing briefly in summer to feed and grow before freezing again, repeating this cycle until it has accumulated enough resources to pupate and complete its metamorphosis. When spring warmth returns, these insects thaw. Their metabolism resumes. They continue, without apparent consequence, from exactly the point at which the cold interrupted them. In the frozen quiet of a winter stem or a patch of leaf litter, something is alive. It is just waiting, as it has learned to wait, for the world to warm. The polar bear is the largest land predator on Earth, and in the landscape it inhabits, sea ice, frozen coastline, the white expanse of the Arctic, it appears at a glance to be perfectly white. The fur matches the snow. The bear moves across ice and is sometimes difficult to see even at moderate distances. The stars are out. But beneath that white fur, the polar bear's skin is black. Dark skin absorbs heat from the sun more efficiently than light skin, and in an environment where every degree of warmth matters, this is not a trivial advantage. The fur itself is not actually white. Each individual hair is transparent and lacks pigment entirely. The whiteness is an optical effect, produced by the way the translucent hairs scatter and reflect incoming light, in the same way that snow appears white despite being made of transparent ice crystals. The fur is also extraordinarily dense. Polar bears have both a dense undercoat and longer guard hairs, creating a layered insulation system that retains heat even when wet, a property that matters for an animal that spends significant time swimming in Arctic waters. The fat layer beneath the skin, which can exceed 10 centimeters in thickness, provides additional insulation and serves as an energy reserve during the long periods when hunting is unproductive. The paws are large, distributing the animal's weight across snow and ice, and the surfaces of the paw pads carry small, soft bumps that improve grip on slick surfaces. The claws are short and curved, suited to gripping prey rather than climbing. From the black skin absorbing warmth at the base to the white fur scattering light at the surface, the polar bear is a study in the accumulated precision of adaptation. The waves are low. They drift with the current, trailing their tentacles through the water, pulsing slowly and with an unhurried regularity that seems almost meditative. Jellyfish are among the oldest animals on Earth. Their basic body plan has been drifting through the oceans for over 500 million years, and they reproduce in ways that reflect their ancient, unconventional biology. Most familiar animals reproduce sexually, combining genetic material from two parents to produce offspring. Jellyfish do this too, in the adult medusa stage that we recognize as a jellyfish. But they also do something else. In their earlier polyp stage, a small sessile form that attaches to hard surfaces on the sea floor, jellyfish can reproduce asexually, producing new individuals by budding or by a process of fragmentation in which portions of the polyp separate and develop independently. A single polyp can generate multiple medusae through a process called strobylation, essentially dividing itself along a series of horizontal planes to release a stack of juvenile jellyfish one by one. Some adult jellyfish can also fragment under certain conditions, with pieces of the bell or tentacle regenerating into complete new individuals. The jellyfish that combines all of these capacities most remarkably is Turritopsis dohrnii, the immortal jellyfish introduced at the beginning of this journey, which can revert entirely to its polyp stage and begin again, sidestepping the usual biological progression toward death in a way that no other known animal manages quite so completely. In the shifting tides, jellyfish drift between one form and many, between individual and colony, between ending and beginning. And then, as the water moves, as a wave breaks, or your hand trails through the shallows, the ocean lights up. Ancient, patient, and endlessly renewable. You are at this moment emitting light. Not the warmth of infrared radiation, which all warm-bodied creatures release, but actual visible light, photons in the range of wavelengths that the human eye is designed to detect. The glow is extraordinarily faint, roughly 1,000 times too dim to be seen under ordinary circumstances, but it is real and it has been measured. The source is a byproduct of the metabolic activity constantly occurring in your cells. As biochemical reactions proceed, as electrons are transferred, as molecules are oxidized, as the ordinary chemistry of cellular life unfolds, a small proportion of the energy released takes the form of photons rather than heat. These photons pass through the skin and into the surrounding air, where they can be detected by instruments of sufficient sensitivity. Japanese researchers using cameras capable of detecting single photons documented this human bioluminescence in a series of experiments published in 2009. They found that the intensity of the glow fluctuates across the day, lowest in the early morning and highest in the late afternoon, following a rhythm that corresponds broadly to the body's metabolic activity. The face tends to emit more light than other body surfaces, perhaps reflecting the higher density of metabolically active tissue in the skin and underlying structures there. Not with reflected moonlight. The glow is not a product of body heat. It is not infrared. It is cold light produced by chemistry, in the same fundamental category as the light of a firefly or the shimmer of a bioluminescent wave, just many orders of magnitude fainter. You shine. Not in any way you could see, and not in any way you could feel. But tonight, as you rest, your body is quietly producing light, one photon at a time. Section 81. Fish that walk on land. The boundary between sea and land is, for most fish, absolute. Water sustains them. Air does not. The transition that vertebrate ancestors made from ocean to land, roughly 375 million years ago, is recorded in the fossil record as one of the most consequential events in the history of life. A crossing that gave rise eventually to every amphibian, reptile, bird, and mammal that has ever lived on the surface of the Earth. Not with any light from above, but for a handful of living fish species, that boundary remains, in a limited but genuine sense, crossable. The mudskipper, found in the mangrove swamps and tidal mudflats of West Africa, South Asia, and the Indo-Pacific, spends a significant portion of its life out of water. It moves across mud and vegetation using its pectoral fins as crutches, swinging its body forward in a motion that resembles, at low speed, a kind of ungainly gallop. It breathes through its skin and the lining of its mouth, provided those surfaces remain moist, an arrangement that requires it to periodically roll in mud or return briefly to water. Some species climb the roots and lower trunks of mangrove trees to bask or to escape aquatic predators. The climbing perch of South and Southeast Asia can move across land by pushing with its gill covers and fins, traveling distances of several hundred meters when water sources dry up or when it detects a more suitable body of water nearby. It breathes air directly into a specialized chamber adjacent to its gills. These fish are not becoming land animals. They are not in the process of any observable transition. They are simply fish that have expanded the boundaries of what fish can do, demonstrating that the line between water and land, even now, is not as fixed as it appears. From within, in the low-lying forests and grasslands of South America, where the wet season can bring weeks of flooding that raises water levels across vast areas of flat terrain, fire ants face a challenge that would defeat most small animals entirely. Their nests built in the soil are inundated. The ground disappears beneath water. There is nowhere to go, except across the surface of the flood itself. Within minutes of rising water reaching the colony, fire ants begin to assemble. Workers link their bodies together using mandibles and adhesive pads on their legs, forming a living mat that spreads across the water's surface. The queen, the larvae, and the eggs are moved to the center and uppermost layers of the structure, protected within the raft, while the workers forming its base and edges remain submerged or at the waterline. The raft floats because of the way the ants' bodies interact with water. The hydrophobic nature of their exoskeletons combined with the air trapped between the tightly packed bodies, creates sufficient buoyancy to support the entire structure. In laboratory experiments, researchers have found that fire ant rafts can remain afloat for weeks without disintegrating. This is bioluminescence, produced by microscopic living organisms called dinoflagellates that drift in vast invisible clouds near the ocean's surface. The structure is not rigid. It flexes and adjusts continuously in response to disturbance. If a portion of the raft is pushed under, the ants in that region paddle and redistribute until equilibrium is restored. The raft moves with the current, and if it contacts a surface, a tree trunk, a branch, the bank of higher ground, the ants at the leading edge begin to climb immediately, and the colony transfers itself to solid ground. No individual ant coordinates the raft's construction. The structure emerges from the simultaneous response of thousands of individuals to the same stimulus, rising water beneath their feet, each following the same simple rules, each contributing one body to the whole. Reach down and cup a handful of seawater from the ocean's surface. Hold it for a moment. Look at it. It appears clear, or perhaps faintly blue, or slightly green depending on where you are. It appears, to the naked eye, to contain almost nothing. When disturbed by movement, they release a brief flash of cold blue-green light, a chemical reaction contained within their tiny bodies, the same fundamental process that makes a firefly glow on a summer evening. It contains thousands of living organisms. A single milliliter of surface seawater, less than a quarter of a teaspoon, can hold between 100,000 and 1 million bacteria, tens of thousands of viral particles, thousands of phytoplankton, and the larvae or eggs of animals from dozens of different species. Some of these organisms are so small that millions of them could rest on the period at the end of this sentence. Others are large enough to see if you know what to look for. A shrimp larva, a copepod, a chain of diatoms linked end to end in a structure that catches the light. Under a microscope, a drop of seawater reveals a world of extraordinary variety. Diatoms, single-celled algae encased in intricate silica shells of geometric precision, drift alongside radiolarians whose skeletons are lace-like spheres of crystalline beauty. Cyanobacteria that are among the most abundant photosynthetic organisms on Earth drift invisibly past organisms that consume them. Each of these microscopic lives is metabolizing, reproducing, responding to its environment, engaging with the organisms around it in relationships of predation and competition and chemical communication that we have barely begun to map. The ocean is not a body of water containing life. It is a body of life that contains water. In certain places, Mosquito Bay in Puerto Rico, the lagoons of the Maldives, sheltered coves in New Zealand, this phenomenon reaches a scale that transforms the coastline entirely. And in every handful of it, a universe is suspended, going about its business, entirely indifferent to being held. In the hydrothermal fields of Yellowstone National Park, pools of water bubble and steam at temperatures approaching or exceeding the boiling point, colored vivid yellows and oranges and deep reds by the communities of microorganisms living within them. In the Danakil Depression of northeastern Ethiopia, one of the hottest and most geologically active places on the surface of the Earth, acidic pools shimmer in colors that seem to belong to another planet entirely. These are not sterile environments. They are teeming with life. The organisms that inhabit these extreme environments are called extremophiles, and among them the thermoacidophiles, microbes that thrive in conditions of both high temperature and high acidity simultaneously, represent perhaps the most remarkable adaptation to hostile conditions that biology has yet produced. Some species are metabolically active at temperatures above 100 degrees Celsius. Others survive in fluids with pH values approaching 0, roughly equivalent to the acidity of battery acid. Their survival depends on cellular machinery that has been fundamentally redesigned by evolution. Their proteins are stabilized by additional chemical bonds that prevent them from unfolding at high temperatures. Waves glow as they break. Their cell membranes are composed of lipids with structures that remain functional and selectively permeable at temperatures that would dissolve the membranes of ordinary cells entirely. They draw their energy not from sunlight, but from the chemical reactions between the minerals dissolved in the hot, acidic water and the gases rising from below, a form of chemosynthesis that requires no light, no oxygen, and no organic matter from other organisms. These microbes may closely resemble the earliest forms of life on Earth, and they remind us that wherever chemistry permits energy to flow, life will find a way to intercept it. Every evening, as the light fades and the process of photosynthesis slows and finally stops, a plant faces a problem that is, in its essentials, a matter of resource management. During the day, photosynthesis has produced sugars, which the plant has been converting into starch and storing. These starch reserves are the plant's only source of energy through the night. There is no sunlight to drive new production until dawn, and the plant's cellular processes do not pause simply because the light has gone. Respiration continues. Growth continues. The maintenance of living tissue continues. Swimmers leave glowing trails behind them. All of it draws on the starch reserve. The problem is calibration. Consume the reserve too quickly, and the plant exhausts its energy supply before dawn, potentially stunting growth or damaging tissue. Consume it too slowly, and energy that could have supported growth goes unused. Researchers studying Arabidopsis, a small flowering plant widely used in botanical research, discovered that plants manage this problem with a precision that is genuinely surprising. The rate at which starch is consumed during the night corresponds closely to the amount of starch available divided by the number of hours remaining until dawn. The plants, in other words, are performing something functionally equivalent to division, computing a consumption rate that will deplete the reserve at almost exactly the moment the sun rises. The mechanism is molecular rather than mathematical. Chemical signals encoding information about starch concentration interact with signals encoding information about the circadian clock, producing an output that regulates the rate of starch breakdown accordingly. No brain performs this calculation. The sea becomes something that no photograph quite manages to capture. No awareness oversees it. A plant in the dark is doing arithmetic, and doing it accurately enough to reach dawn with almost nothing left. An owl launches from its perch in the darkness of an old forest, crosses 30 meters of open air, and drops onto prey in the leaf litter below. A mouse, a volley, a small creature moving through the dark. The owl makes almost no sound in transit. This silence is not accidental, and it is not simply a consequence of the owl flying slowly or carefully. It is the product of a set of feather adaptations so precisely tuned to the suppression of aerodynamic noise that engineers studying owl flight have spent decades attempting to understand and replicate them. The leading edges of an owl's primary feathers, the large flight feathers at the wingtip, carry a comb of stiff, hair-like extensions called flutings or fimbriae, which break up the turbulent air that would otherwise produce a rushing sound as the wing moves forward. The trailing edges of the same feathers are soft and fringe-like, further diffusing air that passes over them. The entire upper surface of the wing is covered in a dense, velvety layer of fine structures that absorb sound at the frequencies produced by flight. Bioluminescence in the ocean serves many purposes. The result is a wing that moves through air without producing the turbulence that generates noise in the wings of other birds. The sound of an owl in flight is below the threshold of human hearing at distances of more than a few meters, and crucially, below the threshold of the hearing of the small mammals it hunts. The owl does not merely approach its prey silently. It approaches in a silence so complete that the prey's own acute hearing offers no warning. In the dark, on soft wings, the owl moves through air as though the air itself does not notice. In the shallow freshwater streams and ponds of much of the world, moving with the unhurried drift of something that has no need to be anywhere quickly, planarians go about their existence. They are flatworms, thin, soft-bodied, typically no more than a few centimeters long, and they are, in proportion to their size, among the most regeneratively capable animals known to science. If a planarian is cut in two, neither half dies. The head end regenerates a new tail. The tail end regenerates a new head, complete with a brain, eyes, a digestive system, and a fully functional nervous system. Some creatures use it to confuse predators. Both halves become complete independent worms, genetically identical to the original and to each other within a matter of days to weeks, depending on the species and conditions. The mechanism underlying this regeneration is a population of cells called neoblasts, which are distributed throughout the planarian's body and retain the capacity to develop into any cell type the animal possesses, a property called pluripotency that is normally associated in most animals only with embryonic cells. When tissue is lost, neoblasts migrate to the wound site, proliferate, and differentiate into whatever cell types are required to rebuild the missing structures. Researchers have pushed the limits of this system experimentally, cutting planarians into numerous fragments and observing that pieces as small as 1/300th of the original worm can regenerate into a complete individual, provided the fragment contains at least a small number of neoblasts. The planarian does not experience its fragmentation as a crisis. It experiences it, to whatever extent it experiences anything, as an inconvenience, one that it resolves quietly and completely by simply growing back. In cities across much of the world, pigeons are so familiar as to be effectively invisible. They move through plazas and train stations and park benches with a casualness that suggests complete indifference to the human activity around them. They are, by common assessment, among the most ordinary of urban animals. They are watching you. Others use it to lure prey. Research conducted over the past 2 decades has established that pigeons can recognize individual human faces with a reliability and persistence that is difficult to reconcile with the casual contempt in which they are generally held. In one series of experiments, 2 researchers of different appearance spent time in the same location, one feeding the pigeons, one chasing them away. After a period of conditioning, the pigeons consistently approached the feeder and avoided the chaser, even when the 2 researchers swapped their clothing entirely. The pigeons were not using clothing as their cue. They were using faces. Further research has shown that pigeons can classify photographs of human faces by individual identity, sorting images they have never seen before into categories based on facial features with an accuracy significantly above chance. They can distinguish between paintings by different artists, between written words and non-words, between the emotional expressions on human faces. Their visual memory appears to be both precise and durable, retaining information about specific individuals over periods of months. In a city where the same pigeons may occupy the same plaza for years, this memory is not trivial. The birds have accumulated detailed records of the humans who share their space. Some communicate with it or use it to vanish against the faint light filtering down from above. The pigeon on the ledge above you has, in all likelihood, already made its assessment. Open any standard account of the human genome, and you will find it described as containing approximately 20,000 protein-coding genes, the sequences that carry the instructions for building the proteins your body is made of and depends upon. These sequences account for roughly 2% of the total DNA in your cells. A further 8% consists of sequences derived from ancient viruses. These are the remnants of retroviruses, a type of virus that, rather than simply hijacking a cell's machinery and then leaving, inserts a copy of its own genetic material directly into the host cell's DNA. If a retrovirus manages to infect a reproductive cell, a sperm or an egg, that insertion becomes heritable. It is copied into every cell of every subsequent generation. It becomes, in the most literal sense, part of the genome. Over millions of years, as these ancient viral sequences accumulated mutations and gradually lost the ability to produce functional viruses, most of them became silent, present in the genome but not expressed, inert remnants of infections so ancient that the viruses responsible no longer exist in any other form. But not all of them remained silent. But here, in the shallows, on a warm and windless night, it simply glows. Some were co-opted by evolution into new functions. Sequences derived from ancient retroviruses are now known to be involved in the regulation of gene expression during embryonic development. One viral protein is essential to the formation of the placenta in humans and other mammals, the very structure that allows the mammalian reproductive system to function as it does. Others appear to play roles in the development of the brain. The boundary between self and other, between human and not human, dissolves somewhat when examined at the level of the genome. You are not simply a human being. You are a human being who carries, written permanently into every cell, the molecular record of ancient encounters between your ancestors and organisms that no longer exist. Encounters that left you, in ways still being discovered, different from what you would otherwise have been. In the Malheur National Forest in eastern Oregon, beneath a forest of conifers on the slopes of the Blue Mountains, lives the largest known organism on Earth. It has no name that most people would recognize. It produces no flowers, no fruit, no leaves. A quiet reminder that even in the darkest water, life finds a way to shine. It makes no sound and moves no part of itself in any direction. And the vast majority of it is invisible. It is a fungus. A single individual of the species Armillaria ostoyi, the honey fungus, whose mycelium, the network of thread-like filaments through which fungi grow and feed, spreads through the soil across an area of approximately 9 square kilometers. Every filament in that network is genetically identical, connected to the others, part of a single continuous organism. Its age is estimated at somewhere between 2,000 and 8,000 years. Some researchers have suggested it may be significantly older. Above ground, the fungus expresses itself modestly, as clusters of small honey-colored mushrooms that appear on the surface of the soil or at the bases of trees in autumn. These mushrooms are the reproductive structures, the equivalent of flowers, temporary, seasonal, and representing only a tiny fraction of the organism's total mass. Below ground, the mycelium extends through the soil in all directions, colonizing the roots of living trees, extracting nutrients from them, and in some cases killing them, creating the gaps in the forest canopy that give the landscape above its particular character. You may feel right now like a single self. The largest animal, the blue whale, is approximately 30 meters long. The largest plant, the Pando aspen colony, covers approximately 43 hectares. This fungus covers 900. Beneath the forest floor in Oregon, something ancient and enormous is going quietly about its existence, as it has been doing since before most of what we think of as history began. Inside a beehive, the flow of information is constant. The famous waggle dance, a figure-8 movement performed by a returning forager on the surface of the honeycomb, encodes the direction and distance of a food source with a precision that researchers spent decades decoding. Other bees watch the dance and use the information it contains to navigate to flowers they have never visited. But bees can learn from observation in ways that go beyond their own instinctive behavioral repertoire. In a series of experiments conducted by researchers at Queen Mary University of London, bumblebees were presented with a task entirely outside their normal experience, rolling a small ball into a circular target to receive a sugar reward. The task was not one that any bee would encounter in the wild, and it had no obvious connection to any behavior bees perform naturally. One body, one mind, one quiet presence lying still in the dark. Bees that observed a trained demonstrator completing the task learned to perform it themselves significantly faster than bees that had to discover the solution independently. More remarkably, bees that watched the demonstration did not simply copy what they had seen. They innovated, finding more efficient methods of completing the task than the ones they had observed. This is social learning of a kind that biologists had not expected to find in insects. The ability not merely to imitate, but to observe, extract the underlying principle, and improve upon it. The implications extend beyond the laboratory. If bees can learn from one another and build upon what they observe, then knowledge, real, transmissible, improvable knowledge, may exist in the hive in a form more sophisticated than we had assumed. The waggle dance was always remarkable. It turns out it may be only the beginning of what bees are capable of telling one another. Sleep, for most land-dwelling animals, is a period of vulnerability. To sleep is to reduce awareness of the surrounding environment, to lower responsiveness to threat, to surrender a portion of vigilance in exchange for the restorative processes that sleep provides. On land, this trade-off is manageable. A sleeping animal can find cover, can rely on stillness and concealment. But you are not alone. In the open ocean, there is no cover. There is no stillness. There is current and predator and the constant need to maintain position in 3 dimensions. And yet the animals that live there must sleep. Some fish, including certain species of parrotfish and wrasse, exhibit patterns of brain activity during rest periods that resemble the slow-wave sleep seen in mammals and birds, a reduced rhythmic neural activity associated with restorative processes. Parrotfish rest motionless near the reef at night, sometimes secreting a mucus cocoon around themselves, and can be approached more closely during these rest periods than when active. Dolphins and porpoises sleep unihemispherically, resting one hemisphere of the brain while the other remains active, then switching, allowing them to continue breathing consciously, maintain directional awareness, and remain responsive to their environment even as half their brain undergoes sleep. They have been observed swimming slowly in tight circles during these rest periods, the open eye oriented outward. Sperm whales rest in a manner that appears to involve both hemispheres simultaneously, floating vertically near the surface in groups, barely moving, in a state of deep sleep that lasts for short periods before the group collectively rouses. The ocean does not pause. You have never been alone. And so the animals that live in it have found ways to carry sleep with them. Partial, adapted, always watchful, woven into the rhythms of a world that never quite goes still. In the forests and scrublands where chameleons live, the first thing most people want to know is how they change color. The second, usually following quickly, is why. The answer to the second question is more interesting than the answer to the first, and it is not primarily camouflage. Chameleons do use color change to blend into their surroundings under certain circumstances. But the more immediate and frequent drivers of color change are internal, shifts in the animal's physiological and emotional state that are broadcast outward through the skin for other chameleons to read. A male chameleon encountering a rival displays a rapid intensification of color, bright yellows, vivid blues, deep reds appearing across the body in patterns that signal aggression and competitive status. A subordinate male, recognizing a dominant one, may respond by fading toward darker, more muted tones, a visible declaration of submission that can forestall physical conflict. A female ready to mate displays receptive coloration. A female that is not receptive, or that is already pregnant, displays patterns that signal rejection. Your body is home to more microbial cells than human ones. The mechanism underlying these displays involves not pigment, but structure. Chameleons possess cells called iridophores that contain arrays of nanocrystals. When the chameleon relaxes or tenses the tissue in which these cells are embedded, the spacing between the crystals changes, altering the wavelengths of light they reflect. The shift from blue to yellow to red requires no new pigment to be synthesized. It requires only the rearrangement of crystals already present, a change that can occur in seconds. The chameleon's skin is a display screen. What it shows is not always a picture of the background. More often, it is a picture of the animal's inner world, rendered in color, broadcast to those nearby, and read with an accuracy that makes the need for words entirely unnecessary. Surrounding the Earth, generated by the movement of molten iron in the planet's outer core, is a magnetic field that extends from deep within the planet out into space for tens of thousands of kilometers. It deflects the solar wind. It shields the surface from cosmic radiation. Trillions of bacteria, viruses, fungi, and archaea live on your skin, in your mouth, across the surfaces of your lungs, and in dense, complex communities deep within your gut. And for hundreds of millions of years, long before any human hand held a compass, it has been guiding living things across the surface of the planet. Sea turtles navigate by it. A female loggerhead turtle, hatching on a beach in Florida, imprints on the specific magnetic signature of that location and carries that information for decades, through migrations that may take her across the Atlantic and back, until she returns, sometimes to within meters of the beach where she hatched, to lay her own eggs. The magnetic map she carries is precise enough to serve as an address. Salmon use it to find their way from open ocean back to the specific tributary in which they were born, combining magnetic information with olfactory cues to achieve a precision of navigation that continues to astonish researchers. Migratory birds, robins, warblers, white-throated sparrows, appear to see the magnetic field, or something related to it, as a visual effect overlaid on their normal vision, mediated by light-sensitive proteins in their eyes that are disrupted when the relevant wavelengths of light are blocked. Even some bacteria contain chains of magnetite crystals that align with the field, orienting the cells along magnetic lines in ways that appear to influence their movement through sediment. The field is everywhere. It has always been everywhere. And life, for as long as life has been complex enough to navigate, has been reading it, following invisible lines across the surface of the world, guided home by forces that most of us will never directly perceive. Together, they form what scientists call the microbiome, and it is not simply along for the ride. And so we come quietly to the end of our journey through this extraordinary world. We have traveled far tonight without moving at all. We have visited the bottom of the ocean, where life first began in the warmth of ancient vents. We have drifted with jellyfish in sunlit water and descended into the permanent dark, where anglerfish carry their own light. We have stood in the stillness of forests, where trees are speaking to each other through the soil, and at the edge of Antarctic ice, where penguins offer pebbles with the gravity of a vow. We have looked briefly at what you are, 30 trillion cells in constant collaboration, 8% ancient virus, bones built from the remnants of dead stars, a body that glows in the dark and hums with electricity and breathes in rhythm with an ocean you may never visit. Through all of it, one truth has surfaced again and again, quietly and without insistence. Life does not give up. It has been frozen, boiled, irradiated, starved, buried, fragmented, and nearly erased 5 times over by events of planetary scale. And each time it has returned. Your microbiome helps break down the food you eat into forms your body can absorb. Not the same, never quite the same, but continuous. Adapted, carrying forward something unbroken from whatever came before. You are part of that continuity, not separate from the wood frog that freezes and thaws, or the tardigrade that drifts through space in dormancy, or the aspen colony that has been a single organism since before the last ice age. You are made of the same fundamental chemistry, shaped by the same long process, sustained by the same ancient rhythms of carbon and water and light. The coral is spawning somewhere tonight, drawn upward by the full moon. A whale song is crossing an ocean in the dark. A seed that has been waiting in the ground since before you were born is measuring the warmth of the soil and finding it at last sufficient. And you are here, breathing slowly, your cells going quietly about their work, your borrowed stardust resting for a few hours before the next day begins. It trains your immune system to recognize threats. Let your breath slow now. Let your thoughts grow soft and drift like spores on still air. You are part of something very old and very large and quietly, persistently, extraordinarily alive. Sleep well. And thank you for dreaming with us. It produces vitamins your cells cannot make on their own. It communicates with your nervous system in ways that researchers are only beginning to understand, influencing mood, behavior, and even the quality of your sleep. Some of these microbes have been with your family line for generations, passed down from parent to child, shaped over time into something almost uniquely yours. No two microbiomes are exactly alike, just as no two fingerprints are. So the next time you feel entirely alone, consider this. You are, in the most literal biological sense, a community. A world within a world. A living ecosystem that breathes and moves and rests. You are not one thing. You are many, all working quietly together. In the dense rainforests of South America, army ants move in vast flowing columns across the forest floor. Hundreds of thousands of individuals traveling together in a living river that rarely stops for anything. But sometimes the path ends. A gap opens. A branch falls. A stream interrupts the route. And when it does, the ants build. One ant anchors itself to the edge. Another grips its body. Then another and another, until a living structure spans the gap. A bridge made entirely of ants, linked leg to leg and jaw to jaw. Flexible enough to sway with the movement of the forest, but strong enough to bear the weight of the colony crossing above. Thousands of individuals may participate. The bridge adjusts in real time, widening where traffic is heaviest, narrowing where it is light. When the last ant has crossed, the bridge quietly disassembles itself, each component rejoining the march. No architect designed this. No leader issued the command. Each ant responds only to the ants immediately around it, following simple chemical and physical cues. And from those local, individual responses, something remarkable emerges. A structure, a solution, a bridge that appears precisely where it is needed and vanishes when it is not. In the forest, intelligence does not always live in a single mind. Sometimes it lives in the space between 10,000 of them. In the dim, shifting world of the ocean floor, the octopus moves like something from a dream. It changes shape. It changes color. It watches with eyes that feel somehow too knowing for an invertebrate, too deliberate for a creature without a backbone. And beneath all of that, something stranger still. Octopuses have the ability to edit their own RNA, the molecular instructions that translate genetic code into the proteins their bodies are built from. Where most animals are locked into the genome they were born with, octopuses can make real-time adjustments to how their genes are expressed. They can alter the behavior of their neurons, fine-tune their responses to temperature and stress, and modify aspects of their biology on the fly, in ways no other animal is known to do quite so extensively. This is not the same as changing their DNA. The underlying code remains. But the way that code is read, the way it is interpreted and acted upon, can shift. It gives them a flexibility that goes beyond anything their genome alone would suggest. We do not yet fully understand why they do this, or how the process is controlled, or what it ultimately means for our understanding of genetics and adaptability. The octopus offers no easy answers. It simply drifts, watches, and edits itself quietly in the dark, one more mystery wrapped inside a creature that already seemed to come from somewhere else entirely. There is a test that researchers use to ask, as carefully as science allows, whether an animal knows it exists. A mark is placed on the animal's body, somewhere visible only in a mirror. If the animal, upon seeing its reflection, reaches for the mark on itself rather than on the image in the glass, it suggests something profound. It suggests the animal understands that the reflection is not another creature. It is them. Very few species have passed this test. Great apes, chimpanzees, gorillas, orangutans recognize themselves. So do bottlenose dolphins, who approach mirrors with what can only be described as curiosity about their own appearance. Elephants have been observed standing before mirrors and touching marked spots on their own faces with the tips of their trunks. Eurasian magpies, birds with brains structured entirely differently from mammals, have passed as well. More recently, certain fish and possibly manta rays have shown signs of the same awareness. None of this means these animals experience the world as we do. The inner life of a dolphin or an elephant remains in most of its detail beyond our knowing. But something is there. Some flicker of recognition. Some boundary between self and world that these animals, in their own way, have drawn. The capacity to know that you exist may be older and more widespread than we ever imagined. Out across the tree of life, in minds built from entirely different materials, something looks back. In the last 500 million years, the world has ended. Not once, not twice, 5 times. Each mass extinction arrived differently. A volcanic age that darkened the skies for centuries. An asteroid that struck with the force of 1 billion nuclear detonations. Shifts in ocean chemistry so severe that the seas themselves became hostile to the creatures living in them. Each time, the majority of species on Earth vanished. Each time, the silence that followed must have been immense. The most devastating of these events, the Permian extinction, which occurred roughly 250 million years ago, erased more than 90% of all marine species and nearly 3/4 of those living on land. The oceans nearly emptied. The forests nearly disappeared. By any reasonable measure, life should have been finished. It was not finished. From whatever survived, the small, the hidden, the organisms tucked into crevices and deep water and the margins of a ruined world, life rebuilt itself. Slowly, over millions of years, new species emerged. New ecosystems assembled. New forms of complexity arose that had never existed before. We are part of that story. Every living thing on Earth today descends from survivors. From creatures that endured the darkness and the cold and the slow return of light and found a way to continue. The thread has never broken. Not once in 500 million years. That, perhaps, is the most quietly astonishing fact of all. In the shaded undergrowth of tropical forests, one of nature's most unsettling dramas plays out in near-perfect silence. A carpenter ant goes about its work, foraging, climbing, following the chemical trails of its colony. Then something changes. Its movements become erratic. It wanders away from the paths it knows. It climbs, seemingly compelled, up the stem of a plant, higher than ants ordinarily go. It finds a leaf at a very specific height, not too high, not too low, and clamps its jaws around the central vein with a grip it will never release. There it dies. And from the back of its head, a slender stalk emerges, growing upward through the forest air until it releases a cloud of spores that drift down onto the ants moving below. The orchestrator of all of this is Ophiocordyceps unilateralis, a parasitic fungus that has spent millions of years perfecting its hold over its host. Once inside the ant's body, it spreads through the tissues, releasing chemical compounds that alter behavior with extraordinary precision. The ant does not simply act strangely, it acts in exactly the way the fungus requires. Scientists still do not fully understand the mechanism. The fungus does not appear to invade the brain directly, and yet it controls behavior with a specificity that continues to astonish researchers. In the quiet forest, the smallest organisms sometimes wield the deepest power. Look up at the sky. Look out at the ocean. Watch a morpho butterfly cross a shaft of sunlight, or a peacock spread its tail in the afternoon. Blue seems to be everywhere. And yet, in the living world, true blue is extraordinarily rare. Almost no animal or plant produces blue pigment. The chemistry required is simply too complex, too costly for evolution to have arrived at it more than a handful of times. The blue we see in nature is, in most cases, not pigment at all. It is architecture. The feathers of a blue jay, the wings of a morpho butterfly, the iridescent throat of a hummingbird, all of these achieve their color not through dye, but through microscopic physical structure. Tiny repeating patterns on the surfaces of scales, feathers, or shells bend and scatter incoming light in such a way that only blue wavelengths are reflected back to the eye. Change the angle and the color shifts. Wet the surface and it may disappear entirely. This is called structural coloration. It is, in the most precise sense, an optical illusion, one produced not by chemistry, but by geometry, by the arrangement of matter at a scale too small to see. Every flash of blue in the wild is a trick of physics. a conversation between light and the tiniest structures life has ever built. And knowing that does not make it any less beautiful. If anything, it makes it more. On the open savanna, where the grass stretches long and gold in every direction, elephants sometimes stop. They stop at the bones of others, at the bleached remains of elephants they may have known, family members, companions, individuals they traveled with for years or decades. And they do not simply pause and move on. They slow. They reach out with their trunks and touch the bones with a gentleness that is difficult to watch without feeling something. They linger, sometimes for a very long time. Elephants have been observed returning to the same remains year after year, examining the same bones with the same careful attention. They have been seen standing guard over dying companions, refusing to leave even when the rest of the herd has moved on. Mothers have carried deceased calves for days, unwilling or unable to set them down. Whatever is happening in these moments, it is not nothing. Elephants form some of the most enduring social bonds in the animal kingdom. They remember. They recognize. And when those they have known are gone, something in them responds to that absence in a way that reaches across the distance between their experience and ours. Their grief may not be identical to our own. The inner world of an elephant is not fully knowable. But it is there, something old and deep and tender, moving quietly through a very large heart. Stretching across the shallow tropical seas of the world are structures so vast they can be seen from space, and every one of them was built by animals smaller than your fingernail. Coral reefs are not rock formations. They are not geological features shaped by pressure or erosion over millions of years. They are cities. Living, breathing, constantly growing cities, constructed grain by grain and layer by layer by creatures called polyps. Each polyp is a small, soft-bodied animal, not unlike a tiny sea anemone. It extends delicate tentacles to catch passing plankton, and it secretes calcium carbonate from its base, building a hard, cup-shaped skeleton around itself. When it dies, that skeleton remains. The next generation builds on top of it. And the next, and the next. Over centuries and millennia, these accumulated skeletons form the reefs we see today. Some stretching for hundreds of miles, rising from the seafloor in towering formations of impossible complexity. Despite covering less than 1% of the ocean floor, coral reefs support an estimated quarter of all marine species. Fish, crustaceans, mollusks, sea turtles, sharks, and thousands of other creatures make their lives within the structure that those tiny polyps build. It is one of the most disproportionate relationships in nature. The smallest of architects, raising the largest of cities, sustaining more life per square meter than almost anywhere else on Earth. It does not have a brain. It does not have a nervous system, or eyes, or any structure that we would ordinarily associate with the ability to think. It is a single-celled organism, visible to the naked eye as a slow-spreading yellow mass on the surface of damp soil or decaying wood. And yet it can solve a maze. In laboratory experiments, researchers placed a slime mold, Physarum polycephalum, at one entrance of a maze, with food positioned at two exits. The organism spread outward in all directions, exploring the available space. Then, gradually, it began to withdraw, pulling back from the dead ends and the inefficient routes, until what remained was a single continuous pathway connecting the two food sources by the most direct route available. No thinking, no planning, no central coordination of any kind. Just chemistry, gradients of signal and response distributed across the entire body of the organism simultaneously, each part reacting to what the parts around it were doing until the whole arrived at an answer. Scientists have since used slime molds to model human transportation systems, finding that the networks these organisms naturally produce bear a striking resemblance to some of the most efficient rail and road systems ever designed. In a quiet patch of forest floor, something without a mind is solving problems that challenge ours. And it has been doing so without fanfare or recognition for hundreds of millions of years. The Amazon rainforest is the largest tropical forest on Earth. It is home to an almost incomprehensible density of life, more species per square kilometer than almost anywhere else on the planet. It regulates rivers, shelters countless organisms, and stores vast quantities of carbon in its wood and soil. But it does something else, something less visible and perhaps even more remarkable. It makes its own rain. Every day, across billions of trees, a process called transpiration draws water up from the soil, through the roots and trunks and branches, and releases it into the air through the leaves as water vapor. This moisture rises into the atmosphere, cools, condenses into clouds, and eventually falls again as rain, which is absorbed by the same trees that produced it. The Amazon generates a significant portion of its own rainfall through this cycle, effectively creating and sustaining its own weather system. The moisture that falls on forests hundreds of miles inland has, in many cases, already passed through a tree. Scientists have begun calling these great flows of aerial moisture flying rivers. When the forest is intact, the cycle is self-sustaining. But when large areas of trees are lost, the cycle weakens. Less vapor means fewer clouds. Fewer clouds mean less rain, and less rain means more trees are lost. The Amazon is not simply a feature of the landscape. It is a living engine of the atmosphere, breathing moisture into the sky above an entire continent. Winter in the northeastern forests of North America is long and absolute. Temperatures fall far below freezing. Snow covers the ground for months. For most small animals, survival means finding warmth underground, or migrating, or burning stored energy through the slow metabolism of hibernation. The wood frog does none of these things. Instead, it freezes. Not metaphorically. Not in a slowed-down, torpor-like state that merely resembles freezing. The wood frog's body drops below 0 degrees Celsius. Its heart stops beating. Its blood stops moving. Ice crystals form in the fluid between its cells. Its organs cease their functions. To any ordinary measure, the animal is dead. And yet it is not dead. The frog's tissues contain high concentrations of glucose, a natural cryoprotectant that prevents the ice from forming inside the cells themselves, where it would cause irreparable damage. The cells are preserved. The DNA is intact. The molecular instructions for being a frog remain exactly as they were. When spring arrives and temperatures rise, the ice melts. The heart restarts. The frog blinks, stirs, and hops away into the wet leaves as though nothing unusual has occurred. Scientists study wood frogs with great interest, hoping to understand what their chemistry might teach us about preserving human tissues, or recovering function after extreme cellular stress. For now, though, the wood frog remains what it has always been, a small, quiet miracle of patience, returning from the cold one heartbeat at a time. Beneath the surface of the sea, in the deep water where light does not reach, and the pressure is immense and the world is very still, a sound is traveling. It began perhaps thousands of miles away. A low, slow, resonant call produced by a blue whale, the largest animal alive. The call is too deep for most human ears to detect without instruments. It is one of the loudest sounds produced by any living creature. And in the right conditions, in the deep sound channel where cold water acts as a kind of acoustic highway, it can travel across an entire ocean basin before it fades. Whales use these calls to find mates, to navigate, and to maintain contact with others of their kind across distances that no other form of communication could span. Each population has its own repertoire, its own dialect, shaped by geography and culture, and the slow accumulation of learned behavior over generations. Humpback whales sing songs of extraordinary complexity, sometimes lasting for hours, that change gradually over the course of months and years. New phrases are introduced, others are dropped. The song evolves, and other whales learn the new version, and it spreads. We do not know everything these songs mean. Much of what passes between whales in the deep ocean remains beyond our understanding. But we know the calls are not random. They are structured, intentional, and shared. In the darkness beneath the waves, across distances almost too great to imagine, the whales are speaking. A seed does not look like much. Held in the palm of your hand, it is small, dry, and still, more like a stone than a living thing. And yet inside it, folded carefully into a protective coat of remarkable durability, is everything required to become a tree, a flower, a fruit, a forest. And sometimes a seed simply waits. Some seeds remain dormant for decades before the right conditions arrive. Others persist for centuries. A few have managed something even more extraordinary. In the 1960s, archaeologists excavating a 2,000-year-old site near the Dead Sea recovered a cache of date palm seeds that had lain in the dry desert earth since the time of ancient kings. Researchers planted one. It germinated. It grew. A tree whose lineage stretched back through 2 millennia of silence had simply been waiting for water and warmth and a reason to begin. Seeds achieve this through a combination of protective chemistry, near total suspension of metabolic activity, and a finely calibrated sensitivity to environmental cues. They do not sprout early. They do not guess. They wait for the precise convergence of moisture, temperature, and light that signals the world is ready for them. There is something deeply calming about this. The idea that life can hold itself in perfect stillness, neither advancing nor retreating, simply present and patient for longer than any human memory reaches. In every seed, a quiet promise. The future held in suspension, waiting for its moment. In the deep ocean, where no sunlight has ever reached, something glows. On a summer lawn, something flickers. In a cave on a forest floor, across the surface of a breaking wave, light appears, cold and living, produced not by fire or electricity, but by life itself. Bioluminescence, the ability to generate light through chemical reaction, has evolved independently at least 40 separate times across the history of life on Earth. It has appeared in bacteria and in jellyfish, in fungi and in squid, in fish and in fireflies, and in the microscopic plankton that makes certain shorelines glow at night. Each time, evolution arrived at the same fundamental solution through an entirely different path. The chemistry is elegant in its simplicity. An enzyme called luciferase acts upon a light-producing molecule called luciferin, releasing energy not as heat but as visible light. Cold, efficient, and extraordinarily precise. Different organisms use this light in different ways. Some lure prey toward them in the darkness. Some warn predators that they are toxic. Some use it to find mates or to camouflage themselves against the faint light filtering down from above. In the deep ocean, where bioluminescence is more the rule than the exception, living light is a primary language, a flickering, drifting vocabulary written in the dark. That evolution has returned to this same solution 40 times, in 40 different lineages, across hundreds of millions of years, suggests something important. In the right conditions, light is not just useful, it is almost inevitable. This is not poetry. It is physics, and it is one of the most quietly staggering things that science has ever established about the nature of your existence. The calcium in your bones was not made on Earth. It was forged in the interior of a star, a massive ancient star that burned for millions of years before exhausting its fuel and collapsing inward in a fraction of a second, then exploding outward in a supernova of almost incomprehensible violence. In the final moments of that explosion, elements heavier than hydrogen and helium were created through the crushing pressure of nuclear fusion and scattered across space in an expanding cloud of gas and dust. Over millions of years, that cloud drifted and cooled and began, under the slow pull of gravity, to collapse again. A new star formed at the center. Around it, rings of dust and debris coalesced into planets. One of those planets, the third from the new star, was small and rocky and, in time, developed oceans. The calcium from that ancient supernova settled into those oceans. It moved through the chemistry of early life. It passed through organisms too small to see, through creatures that swam and crawled and walked across billions of years and billions of generations until it came to rest for now in you. You do not merely live in the universe. You are made of it, assembled from the remnants of something that burned and died long before the earth existed, into a pattern that can breathe and wonder and look up at the stars it came from. When you look at a flower, you see color. Perhaps yellow petals, or white, or a deep and saturated red. You see what the flower shows to eyes like yours, eyes built to perceive a particular range of the light spectrum, the range that falls between red and violet. But a bee sees something else entirely. Bees can perceive ultraviolet light, a portion of the spectrum that lies beyond the violet end of the range visible to humans. And in ultraviolet, many flowers that appear simple or even plain to our eyes are revealed as something entirely different. Patterns emerge. Rings of contrasting color appear around the center of the bloom. Lines and gradients that are invisible to us become vivid guides pointing directly toward the nectar within. These patterns did not appear by accident. They evolved specifically to be seen by pollinators, refined over millions of years in a co-evolutionary conversation between flower and bee. The flower offers a visual signal. The bee follows it. Both benefit. Other insects use ultraviolet vision for different purposes, to navigate by polarized light in the sky, to find mates whose wings carry ultraviolet patterns invisible to predators, to detect the subtle shimmer of water from above. We share this planet with creatures that perceive it in ways we cannot directly experience. The world you see is real, but it is not the whole world. There are colors in the garden that no human eye has ever witnessed, hidden in plain sight, broadcast on a frequency we were simply not built to receive. In the dry, unpredictable expanses of the Australian interior, where rain is unreliable and food can disappear for months at a time, kangaroos have evolved one of the most extraordinary reproductive strategies in the animal kingdom. They can pause a pregnancy. When conditions are harsh, or when a joey already occupying the pouch requires too much of the mother's resources to allow for another, a female kangaroo can suspend the development of a fertilized embryo at a very early stage. The embryo remains viable but dormant, held in a state of biological suspension, waiting. When the pouch becomes available, when the older joey grows independent enough to venture out, or when food and water return in sufficient abundance, the signal is given and the paused embryo resumes its development, continuing as though no interruption had occurred. A mother kangaroo can simultaneously be nursing a large joey outside the pouch, nurturing a younger joey still within it, and carrying a third embryo in suspension all at once. 3 stages of development running in parallel, managed by a biology so finely tuned it can read the state of the environment and respond accordingly. Scientists call this embryonic diapause. It is not unique to kangaroos. A handful of other mammals share a version of the ability, but nowhere is it quite so refined or so essential to survival as in these animals, whose lives depend on matching the rhythms of reproduction to the unpredictable rhythms of a continent. In the coral reef, identity is not fixed. Among the clownfish, small, bright, and famously devoted to their host anemones, social structure is organized around a strict and simple hierarchy. At the top is the dominant female, the largest and most reproductively active individual in the group. Below her is the dominant male. Below him, a collection of smaller, sexually immature fish that wait. If the female dies, the male does not simply find a new mate. He becomes one. Over the course of days or weeks, his body undergoes a complete biological transformation. Hormone levels shift. Gonads restructure. Behavior changes. He becomes female, the new dominant individual at the top of the hierarchy, while the next fish in line steps up to fill the role of male. In other species, the transition runs in the opposite direction. Wrasses and parrotfish live in groups where females may transform into males when the dominant male disappears. Again, the change is not behavioral alone. It is physiological, complete, and permanent. These transitions are not anomalies or errors. They are adaptations, elegant solutions to the challenge of maintaining reproductive capacity in small, isolated social groups, where the loss of any individual could otherwise threaten the group's survival. In the ocean, the boundaries we draw around identity are more permeable than we tend to assume. Life, as it so often does, finds flexibility where we expected rigidity. Every autumn, across much of the northern hemisphere, billions of birds lift into the sky and begin to move. Some travel only a few hundred miles. Others cross continents and oceans, navigating from a summer breeding ground in the Arctic to a wintering site in Africa or South America, then returning the following spring to within meters of where they were born. They do this without maps, without instruments, without anyone to show them the way. One of the mechanisms that makes this possible is magnetoreception, the ability to sense the Earth's magnetic field directly. Some birds carry tiny crystals of magnetite in their beaks or skulls, particles of iron-containing mineral that align with the planet's magnetic field lines like the needle of a compass. Others appear to use specialized proteins in their eyes called cryptochromes, which may allow them to perceive the magnetic field as a visual overlay, a kind of directional tinting across their field of view. This magnetic sense is used alongside other navigation tools: the position of the sun, the pattern of stars, the memorized shapes of coastlines and river valleys. Together, these systems create something more reliable than any single method alone. We have known for decades that birds navigate by magnetism. We still do not fully understand how. The precise mechanism, the exact experience of it, remains just beyond our grasp. Somewhere above you tonight, a bird is crossing the darkness, guided by forces invisible to both of you, following a map written into its body before it ever took flight. Carbon is everywhere. It is in the air you are breathing right now, in the wood of the floor beneath you, in the body of every living thing on Earth, in the stone of ancient seabeds, in the oil locked beneath the surface of the ground, and in the slow exhalations of volcanoes that have been releasing it for billions of years. And it moves. The carbon cycle is the great slow circulation of this element through every corner of the Earth system. You breathe it out as carbon dioxide. A plant nearby absorbs it through its leaves and uses it to build sugars, stems, and roots. An animal eats that plant and incorporates the carbon into its own tissues. When the animal dies, decomposers break it down, returning carbon to the soil and air. Some of it washes into rivers and eventually into the ocean, where it may be absorbed by algae or settled into sediment on the seafloor, or incorporated into the shells of marine animals that will eventually become limestone. Some carbon is locked away for millions of years. Some cycles through living things within days. The ocean breathes it in. Volcanoes breathe it out. The forests hold it and release it and hold it again. This cycle has been running, largely in balance, for hundreds of millions of years. It regulates the temperature of the planet, the chemistry of the oceans, and the composition of the air. It is, in a very real sense, the breath of the Earth itself, slow, vast, and older than anything alive. Before there were forests, before there were oceans full of fish, before there were insects or flowers or bones of any kind, there were builders. They were microscopic. They had no eyes, no limbs, no intelligence in any recognizable sense. And yet they constructed structures that still exist today, scattered across the shallow coastal waters of the world, in places like Shark Bay in Western Australia. Low, rounded formations of layered rock that rise from the seafloor like ancient monuments. These are stromatolites, and they are built by cyanobacteria, photosynthetic microbes that trap and bind fine particles of sediment as they grow, laying them down in thin sheets, layer upon layer, over timescales so long they are difficult to hold in the mind. Fossilized stromatolites have been found that date back more than 3.5 billion years, making them among the oldest direct evidence of life on Earth. But the cyanobacteria were not simply building structures. As they photosynthesized, they were also producing oxygen, releasing it into the water and eventually into the atmosphere, slowly and over immense stretches of time, transforming a world that had never known it into one that breathed it. The living stromatolites that survive today in Shark Bay are direct descendants of those ancient builders, unchanged, unhurried, and still at work. Before any of the rest of life was possible, they were here, laying the groundwork, one microscopic layer at a time. In the warm, clear shallows where dolphins travel in loose and shifting groups, a sound passes between two individuals, a particular sequence of whistles, rising and falling in a pattern unlike any other in the ocean around them. It is a name. Each dolphin develops a signature whistle in the first months of its life, a unique acoustic identity, distinct from every other whistle in the population. Other dolphins learn this whistle. They remember it, and they use it, calling it out in contexts that strongly suggest they are directing their attention to a specific individual, the way you might call across a room to someone you recognize. When researchers in controlled settings played back a dolphin's own signature whistle through an underwater speaker, the animal responded, turning toward the sound, approaching, vocalizing in return, in a way that it did not respond to the whistles of other dolphins. It recognized its own name. Dolphins live in what biologists call fission-fusion societies, groups that are constantly forming, dissolving, and reforming as individuals come and go. In such a social world, the ability to identify specific individuals by name, to remember who they are across separations of days or weeks or longer, is not a curiosity. It is essential. Beneath the waves, in the flickering blue light of shallow water, a system of names is being used and remembered and passed on. Something that we once considered uniquely human turns out to have been spoken in the sea long before we learned to speak at all. Spread across the northern third of Africa, the Sahara is the largest hot desert on Earth. It stretches for nearly 5,000 miles from the Atlantic coast to the Red Sea, a vast ancient landscape of sand and stone and wind where temperatures can exceed 50 degrees Celsius and rain may not fall for years at a time. It was not always this way. Between roughly 6,000 and 11,000 years ago, during a period scientists call the African humid period, the Sahara was green. Monsoon rains, driven by subtle shifts in the Earth's orbital geometry that altered the angle and intensity of seasonal sunlight, penetrated deep into the continental interior. Rivers flowed across what is now open desert. Lakes shimmered in basins that today hold nothing but cracked and dusty earth. Grasslands spread across the dunes. Animals moved through this landscape that would be unrecognizable there today. Elephants, crocodiles, hippos, and giraffes roamed territory that is now lifeless sand. And people lived there too, leaving behind cave paintings on rock walls in the middle of what is now empty desert. Paintings of cattle, of swimmers, of animals that could only have existed in a very different world. Then gradually the rains retreated. The grasslands dried. The lakes evaporated. And the desert returned. Beneath the Sahara today, buried under meters of sand, lie the bones of that vanished world. Ancient lake beds, fossilized tree roots, the traces of rivers that once ran to the sea. The desert remembers, and somewhere in its deep silence, the green world it once was is still there, waiting in the dark beneath the dunes. In the long history of mathematical thought, zero is a latecomer. Ancient civilizations built pyramids and mapped the stars without it. The concept of nothing as a number, as a quantity that can be counted, compared, and reasoned about, is an abstraction that took human cultures thousands of years to fully develop. And yet honeybees appear to understand it. In a series of carefully designed experiments, researchers trained bees to associate smaller quantities with a food reward, teaching them, in essence, that fewer is better. When the bees were then shown a blank card containing no symbols at all, the majority chose it over cards bearing 1, 2, 3, or more symbols. They placed 0 correctly on the numerical scale, below 1, recognizing that none is less than some. This is not simple counting. It is abstract numerical reasoning, the understanding that an absence of quantity is itself a quantity, and that it occupies a specific position in an ordered series. Only a small number of species have demonstrated this capacity, great apes, dolphins, and certain corvids among them. That bees, with brains containing fewer than 1 million neurons, compared to the roughly 86 billion in a human brain, can perform this kind of reasoning is not simply charming. It is scientifically significant. It suggests that the cognitive tools required for abstract thought may be far more widely distributed across the animal kingdom than we have assumed. In the hive, something very small is thinking about nothing, and doing it rather well. In 1995, after an absence of nearly 70 years, wolves were reintroduced to Yellowstone National Park in Wyoming. 14 animals, brought from Canada, were released into a landscape that had forgotten them. What followed was one of the most dramatic demonstrations of ecological interconnection ever documented. The wolves hunted elk as expected, but their effect on the elk went beyond simple predation. The elk changed their behavior. They began to avoid the open river valleys and steep-sided gorges where wolves could corner them, moving instead through terrain that offered more options for escape. And in the valleys the elk had abandoned, something unexpected began to happen. Plants grew. Willows and aspens and cottonwoods, long kept short by constant grazing, began to reach their full height along the riverbanks. As the vegetation recovered, songbirds returned to nest in the new growth. Beavers arrived and built dams, creating ponds that raised the water table and provided habitat for fish, amphibians, and waterfowl. The roots of the recovering trees stabilized the riverbanks, reducing erosion. The rivers themselves began to change course, running straighter in some places, meandering more naturally in others, their channels deepening and their water running clearer. Wolves, simply by being wolves, had changed the shape of the rivers. This phenomenon, in which the presence or absence of a single predator cascades through an entire ecosystem, altering the behavior of herbivores, the growth of plants, the flow of water, and the structure of the land itself, is called a trophic cascade. Yellowstone showed it in motion. Everything is connected. Sometimes the thread runs in directions we never thought to look. For a long time, the idea that plants could respond to sound seemed like the kind of thing that belonged to folklore rather than science. Plants, after all, have no ears, no nervous system, no mechanism, as far as anyone knew, for detecting vibration in the air around them. Then researchers began to look more carefully. In studies published in recent years, certain flowering plants, evening primroses among them, were shown to produce sweeter nectar within minutes of being exposed to the sound of a bee's wing beats. Not the bee's presence, not its scent or the slight movement of air it caused or any visual cue. Just the sound, a specific frequency produced by the vibration of the insect's wings. The flowers achieved this not through ears, but through their petals, which vibrate in response to incoming sound waves, acting as passive acoustic receivers tuned to the frequencies most associated with approaching pollinators. When the right vibration arrives, a signal passes through the plant's tissues, and nectar production increases. The evolutionary logic is clear. A flower that offers sweeter nectar precisely when a pollinator is nearby is more likely to be visited, more likely to be pollinated, and more likely to reproduce. The bee gets a better reward. The flower gets a better outcome. Both benefit from a conversation conducted entirely in the language of vibration. The garden is not silent. It is listening. And in the soft hum of a bee passing between blossoms, the flowers are already beginning to respond. On the surfaces of rocks, on the bark of trees in old forests, on the face of stone walls and the sides of ancient monuments, you will find them, low, crusty patches of color, gray and green and orange and pale yellow, clinging to surfaces that offer almost nothing in the way of nutrition or shelter. These are lichens, and they are not what they appear. A lichen is not a single organism. It is a partnership, a stable, long-term biological alliance between a fungus and a photosynthetic partner. usually a species of alga or cyanobacterium. The fungus provides the physical structure, the outer layers that hold moisture and protect against the elements, the anchoring filaments that grip the rock surface. The alga or cyanobacterium lives within that structure, capturing sunlight and converting it into sugars that feed both partners. Together, they can survive in places where neither could exist alone. Lichens colonize bare rock in the high Arctic and in hot deserts. They grow on the surfaces of Antarctic stones that are exposed to conditions few other organisms can tolerate. They are among the first living things to appear on newly formed volcanic rock, slowly breaking down the surface through the chemistry of their growth, beginning the long process of building soil. The partnership is ancient. Lichens have existed for hundreds of millions of years, and it is not always entirely equal. The fungus shapes and constrains its partner in ways that can resemble control as much as cooperation. But the result endures. Two kingdoms of life woven together into something neither could have become alone. Beneath the sun-baked soils of sub-Saharan Africa, in winding tunnel systems that may extend for hundreds of meters through hard-packed earth, live some of the most biologically unusual mammals on the planet. Naked mole rats are small, nearly hairless, and to most human eyes, profoundly unattractive. They are functionally blind, spending their entire lives underground, and they live in colonies of up to 300 individuals organized around a single breeding queen, a social structure more reminiscent of an insect colony than a mammal society. But it is their relationship with time that makes them truly remarkable. A naked mole rat can live for more than 30 years, an extraordinary lifespan for an animal of its size. A mouse of comparable dimensions lives for 2 or 3. And unlike almost every other mammal studied, naked mole rats do not show an increase in mortality risk as they age. In most animals, the probability of dying rises steadily with each passing year, following a pattern so consistent it has its own name, the Gompertz law. Naked mole rats appear to defy it. Their cells resist the kinds of protein damage associated with aging in other species. They show a remarkably low incidence of cancer, perhaps the lowest of any mammal studied. They tolerate low oxygen environments that would cause brain damage in most other animals, apparently without lasting harm. Scientists studying the biology of aging return to them again and again, searching for the mechanisms that allow these small, strange, underground creatures to age so differently from the rest of us. Whatever their secret is, they are keeping it quietly in the dark. You are not, in any simple sense, a single thing. The body you inhabit is composed of approximately 30 trillion human cells, 30 million individual living units, each performing its own specific role with a precision that no human engineering has ever come close to matching. Muscle cells contract in coordinated sequences to produce movement. Neurons transmit electrical signals across synaptic gaps to generate thought and sensation. Red blood cells carry oxygen from the lungs to every corner of the body and return with carbon dioxide. Immune cells patrol the tissues, identifying and neutralizing threats with a discrimination that can distinguish self from non-self at the molecular level. Each of these cells contains the same complete copy of your genome, the full instructions for building a human being. And yet, each reads only the portions relevant to its function, ignoring the rest with remarkable consistency. And the body is not static. It renews itself constantly. The lining of your gut replaces itself every few days. Your red blood cells are remade every few months. Even your bones, which feel permanent, are being continuously dissolved and rebuilt. The body you inhabit today is not entirely the same body you inhabited a year ago. Through all of this renewal, through all of this constant activity and replacement and coordination, a coherent self persists. Something that thinks and feels and breathes, and right now, in this moment, is quietly at rest. You are not one thing. You are a community, 30 trillion lives working in concert, holding you together. In the 1970s and 1980s, a researcher named Irene Pepperberg began working with an African grey parrot she called Alex. What followed over the next 30 years would quietly reshape the scientific understanding of animal cognition. Alex could identify objects by color, shape, and material. He could count collections of up to 6 items and report the number verbally. He understood the concept of same and different, applying it reliably to objects he had never encountered before. He grasped the idea of absence, what he called none, in a way that placed him among the very few non-human animals known to handle the concept of zero. When he was tired of a task, he said so. When he made an error and was corrected, he sometimes corrected the experimenter in return. In controlled tests across a range of cognitive tasks, Alex performed at the level of a human child between 4 and 6 years of age. African grey parrots are not unique in their intelligence. Keys, macaws, and cockatoos have all demonstrated sophisticated problem-solving abilities in the wild and in laboratory settings. They use tools, navigate complex social hierarchies, and in some cases appear to engage in what can only be described as play. Their brains are structured entirely differently from those of mammals. The regions associated with complex cognition in primates have no direct equivalent in birds. And yet evolution, working from a completely different starting point, arrived at something functionally similar. Intelligence, it turns out, is not one thing. It is a destination that many different roads can reach. More than 70% of the surface of the Earth is covered by ocean. It is the planet's defining feature, the vast, restless body of water that makes Earth visible from space as a blue sphere against the darkness. And most of life is in it. The ocean contains the overwhelming majority of Earth's living biomass. From the sunlit surface waters, where phytoplankton drift in clouds too small to see individually, but vast enough to be visible from satellites, to the abyssal plains 4 miles below, where creatures that have never seen light move in the permanent cold and dark. Life fills the ocean in forms and densities that the land cannot match. A single liter of surface seawater may contain millions of bacteria, thousands of phytoplankton, and the larvae of animals from dozens of different species. A coral reef supports more species per square meter than almost any terrestrial ecosystem. The open ocean, for all its apparent emptiness, is threaded with invisible webs of predation and competition and symbiosis that span entire hemispheres. We have explored only a small fraction of it. The deep ocean, below 200 meters, where sunlight ceases to penetrate, remains less well mapped than the surface of the moon. What lives there, in the trenches and the cold and the absolute dark, is largely unknown. The greatest wilderness on Earth is not a forest or a mountain range or a polar ice sheet. It is the blue expanse that covers most of the world, teeming and ancient and almost entirely beyond our knowing. In a quiet grove in the Fish Lake National Forest in south-central Utah, something grows that does not look remarkable at first glance. A stand of quaking aspens, slender-trunked, pale-barked, their leaves trembling in the slightest movement of air, spreads across about 100 acres of mountain slope. But these are not separate trees. Every trunk in the grove is genetically identical. Every one of them grows from the same vast root system, a single subterranean network that spreads through the soil in all directions, sending up new stems wherever conditions allow. What appears to be a forest of individual trees is, in biological terms, a single organism, one entity that happens to express itself as thousands of trunks. This organism is called pando, from the Latin for eye spread. It is estimated to weigh around 6 million kilograms, making it one of the heaviest known organisms on Earth. And it is thought to be approximately 80,000 years old. 80,000 years! When Pando's root system was already ancient, woolly mammoths still grazed the grasslands of North America. Human beings had not yet reached the continent. The most recent ice age had not yet begun. Individual trunks within the colony live for decades before dying and being replaced by new growth from the same roots. The forest changes constantly at the surface while remaining beneath the ground essentially unchanged. Pando does not hurry. It does not need to. It has already outlasted almost everything that has ever lived on the continent above it, one quiet root at a time. When we think of viruses, we tend to think of illness, of fever and fatigue, of contagion and quarantine, of microscopic particles that invade our cells and turn their machinery toward their own reproduction. The relationship between virus and host, in this framing, is simple and adversarial. But the full picture is considerably more complex. Certain viruses, called bacteriophages, prey not on human cells, but on bacteria, including many of the harmful bacteria that can take hold in the human gut. By keeping bacterial populations in check, these viruses play an active role in maintaining the microbial balance that our health depends upon. More remarkably, approximately 8% of the human genome consists of sequences derived from ancient retroviruses, viruses that infected our ancestors millions of years ago, inserting their genetic material into reproductive cells, where it was copied and passed down through generations. Most of these sequences have been silenced by mutation over time, but some have been repurposed by evolution, co-opted into roles that now serve our own biology. Viral sequences are involved in the formation of the placenta, the structure that allows a mammalian mother to sustain a developing embryo. Others appear to play roles in early brain development and in the regulation of gene expression. Without these ancient viral contributions, human reproduction and development would be, in ways we are still working to understand, different. You are not simply a human being shaped by human evolution. You are, in part, the product of infections that occurred before our species existed, ancient encounters that left their mark permanently in the instructions that make you who you are. 400 million years ago, the oceans of Earth were already full of life. Fish were diversifying into forms we would recognize today. Invertebrates of extraordinary variety moved through shallow seas. The first tentative experiments in land colonization were beginning, as primitive plants crept slowly across the edges of continental margins. And sharks were already there. The shark lineage extends back at least 450 million years, to a time before the first forests, before the first trees, before the first leaf had ever caught the light of the sun. When the great Devonian forests finally appeared, roughly 350 million years ago, sharks had already been swimming for 100 million years. They have since survived every mass extinction event in Earth's history, including the asteroid impact that ended the reign of the non-avian dinosaurs 66 million years ago. Through each of these catastrophes, events that reshaped the entire biosphere, eliminating the majority of species living at the time, the shark lineage persisted. Their basic body plan has remained remarkably consistent across this immense span of time. Not because sharks are simple or unadapted, but because their design was so effective from the beginning that evolution found little reason to depart from it substantially. Cartilaginous skeleton, powerful musculature, acute sensory systems, and an extraordinary sensitivity to electrical fields in the water around them. Today's sharks are not relics. They are the living continuation of a lineage so ancient and so successful that 450 million years of Earth's most dramatic changes have not been enough to end it. In the rocky shallows and kelp forests of the world's cooler oceans, a small and industrious creature goes about its daily business wearing, quite literally, a garden on its back. Decorator crabs are a group of species within the spider crab family that have evolved a remarkable approach to survival. Their shells are covered in small hooked hairs, tiny biological Velcro, designed to grip and hold whatever the crab chooses to attach to itself. And the crab chooses carefully. Moving slowly across the seafloor, a decorator crab selects pieces of algae, sponge, coral fragments, sea anemones, and other organisms from its environment, and presses them deliberately onto its shell, where they catch on the hooked hairs and hold fast. Over time, the crab accumulates a living coat of material drawn from its immediate surroundings, a mosaic of its local environment, worn as camouflage. The disguise is often extraordinarily effective. A crab resting motionless on a rock encrusted with the same species it has attached to itself can be nearly impossible to detect. But the relationship sometimes goes further than simple concealment. Some of the organisms attached to the crab continue to live and grow in place, their own chemical defenses, toxins, unpleasant tastes, or irritating compounds, now extending their protection to the crab as well. When the crab moves to a new environment, it may rearrange its decorations, removing what no longer fits and selecting new material from the surroundings. It is camouflage as curation, survival as slow, deliberate art. We are accustomed to thinking of ourselves as the dominant form of life on Earth. We have built cities and satellites, crossed oceans and mapped genomes. By our own measures, we are extraordinary. And yet, by almost every other measure, the planet belongs to the insects. For every single human being alive on Earth, there are an estimated 200 million insects. They inhabit every terrestrial ecosystem on the planet, from the canopies of tropical rainforests to the frozen edges of the Arctic tundra, from the driest deserts to the wettest swamps. They have been here for more than 400 million years, and they have survived every mass extinction that has occurred in that time. Beetles alone account for nearly a quarter of all known animal species. Ants, taken together, constitute a biomass comparable to that of all humans combined. Termites build structures that regulate their own climate. Bees pollinate the crops that feed us. Flies and beetles and countless others decompose the dead and return their nutrients to the living world. Without insects, most terrestrial food chains would collapse within months. The birds, the reptiles, the small mammals that feed on them would follow. The plants that depend on insect pollination would fail to reproduce. The ecosystems we take for granted would unravel in ways that are difficult to fully imagine. We may have named ourselves the dominant species. But the insects were here first. They are here in far greater numbers, and in all likelihood, they will be here long after we are gone. In the dense, humid rainforests of New Guinea and the islands around it, evolution has produced something that seems, by any ordinary standard, excessive. The birds of paradise, a family of roughly 45 species, have developed courtship displays of such extraordinary complexity and beauty that naturalists who first encountered them in the 16th century refused to believe they were real birds. The skins brought back to Europe by early traders were so bizarre, so extravagant, so unlike anything known, that some concluded the birds must be mythical. They are not mythical. They are the product of millions of years of sexual selection, in an environment where food is abundant enough that males can afford to spend enormous amounts of time and energy on display, rather than survival. Each species has its own choreography, refined over generations into something precise and repeatable. One species fans an iridescent breast shield and sways with hypnotic rhythm. Another hangs upside down from a branch and spreads its plumes into a perfect geometric disc. A third clears a patch of forest floor, arranges objects upon it with careful attention, and performs an elaborate dance at its center. The females watch from nearby perches, sometimes for extended periods, assessing each performance with what appears to be genuine discrimination. They choose the males whose displays are most precise, most vigorous, most complete. What drives this system is the accumulation of small preferences over immense stretches of time. Each generation of females chooses, and each generation of males becomes, incrementally, more extraordinary. The result is a living argument that beauty in nature is not accidental. In a corner of a garden, or along the edge of a windowsill, or stretched between the branches of a tree in the early morning when dew makes it visible, a spider's web catches the light. It looks fragile. It is anything but. Spider silk is, weight for weight, one of the strongest materials found in nature. A thread of spider silk the same diameter as a thread of high-grade steel can withstand significantly more force before breaking. And unlike steel, it is elastic, capable of stretching up to 40% of its length before snapping, absorbing the kinetic energy of a flying insect without shattering. It is simultaneously tough and flexible in a combination that human engineers have spent decades trying to replicate. The silk is produced in specialized glands within the spider's abdomen, extruded as a liquid protein through structures called spinnerets, and drawn into a solid thread by the spider's legs and the tension of the line itself. Different glands produce different kinds of silk: sticky capture silk for the spiral threads of a web, dry dragline silk for the structural frame, soft and cushioned silk for egg sacs, and varieties in between tailored to specific functions. Some spiders produce up to 7 distinct types of silk from a single body. Scientists have been attempting for decades to synthesize spider silk artificially, understanding that a material with its combination of strength, flexibility, and lightness could transform medicine, engineering, and materials science. So far, the spider remains ahead of us. Every morning, quietly and without fanfare, it spins what we cannot yet make. On warm evenings in the meadows and woodlands of the eastern United States, and in similar habitats across much of the world, darkness brings a conversation that has been taking place for millions of years. Fireflies rise from the grass as the light fades, tracing slow arcs through the air, and as they fly, they speak. Each species has its own signal, a pattern of flashes with a specific rhythm, duration, and interval, as distinctive as a signature. Males fly and flash. Females perched in the vegetation below observe, and when they recognize the pattern of their own species, they respond with a timed flash of their own. The exchange continues, drawing the partners closer until they meet. The light is produced in the firefly's abdomen through the same fundamental chemical reaction found in other bioluminescent organisms, luciferase acting on luciferin in the presence of oxygen. But the firefly has evolved exquisite control over this reaction, switching it on and off with a precision measured in fractions of a second. Some species of firefly have developed a darker use for this language. Females of the genus Photuris have learned to mimic the response signals of other species, luring males of those species close and then consuming them, gaining in the process defensive chemicals from their bodies that make the mimics themselves unpalatable to predators. In the soft dark of a summer evening, what looks like simple beauty is also a complex and ancient dialogue, a language of light, read in the dark, by creatures that have been speaking it far longer than we have been watching. In the forests of East Africa, a chimpanzee moving through the undergrowth pauses at a particular plant and strips a bitter, fibrous stem with its teeth. It does not eat the stem for its nutritional value. The taste is unpleasant, the material largely indigestible. It chews slowly, extracting the juice, then discards the pulp. Researchers observing this behavior found that it correlated consistently with the presence of intestinal parasites. The plant selected contained compounds with documented antiparasitic properties. Chimpanzees that consumed them showed reduced parasite loads in the days that followed. This is not an isolated observation. Across a range of primate species, and in a growing number of other animals as well, researchers have documented deliberate self-medication, the intentional use of specific plant materials to address specific health problems. Chimpanzees swallow rough leaves whole without chewing to mechanically dislodge intestinal worms. Capuchin monkeys rub certain plants into their fur, apparently to repel insects. Pregnant lemurs in Madagascar consume plants with known uterine-stimulating properties in the period before birth. The behavior is selective, appearing in response to symptoms rather than as a routine part of diet. It is sometimes learned young animals observed adopting the same remedies used by older individuals in their group. Scientists call this zoopharmacognosy, animal self-medication. It is a field still in its early stages, and what it is beginning to reveal is that the pharmacy of the forest has had customers far longer than it has had human ones. Take a breath. Fill your lungs with the air around you and hold it for a moment. Roughly half of the oxygen in that breath came from the ocean. Not from trees. Not from forests. From the sea, specifically from the vast invisible communities of microscopic organisms called phytoplankton that drift near the surface of the world's oceans, carried by currents across distances that dwarf any land ecosystem. Phytoplankton are photosynthetic. Like plants, they capture sunlight and use it to convert carbon dioxide and water into sugars and oxygen. But they do this at a scale and in quantities that the land cannot match. Spread across the surface of an ocean that covers more than 70% of the planet, these microscopic organisms collectively produce an estimated 50 to 80% of the oxygen in Earth's atmosphere. They also form the foundation of the marine food web. Phytoplankton are consumed by zooplankton, which are consumed by small fish, which are consumed by larger fish, which are consumed by whales and seabirds, and eventually by us. The entire edifice of ocean life, from the smallest krill to the largest blue whale, rests upon the productivity of organisms too small to see individually with the naked eye. Phytoplankton populations respond to temperature, nutrients, and ocean chemistry in ways that researchers are still working to fully understand. They are sensitive and they are essential. Every breath you draw in some part belongs to the sea. The ocean and your lungs are more intimately connected than they may appear. A crow lands on a branch at the edge of a park, holds a walnut in its beak for a moment, then flies to a nearby road and places the nut carefully in the path of approaching traffic. It waits. A car passes over the nut, cracking the shell. The crow waits again for the traffic to clear, then descends and retrieves the kernel. This behavior has been documented repeatedly in multiple cities across several continents. It is not instinct. It is a learned technique, observed in some cases to be passed from one bird to another within a population. Crows are members of the corvid family, which includes ravens, jays, and magpies, a group whose cognitive abilities have, in the past few decades, repeatedly surprised researchers who expected to find intelligence of this kind only in primates. Crows plan for the future, selecting tools in advance for tasks they will not perform until later. They hide food in complex caches, remembering not only where each cache is located, but when each item was stored and whether another crow was watching when they did it, adjusting their caching behavior accordingly to prevent theft. They recognize individual human faces, remember which faces belong to people who have treated them poorly, and have been observed recruiting other crows to harass those individuals. In controlled laboratory settings, corvids have demonstrated causal reasoning, analogical thinking, and the ability to solve multi-step problems requiring the use of tools to obtain other tools. Their brains are small. Their solutions are not. Close your eyes for a moment and imagine a world in which light is irrelevant, in which darkness is not an obstacle but simply a neutral condition, no different from daylight, in which every warm-blooded creature nearby glows with an inner radiance that no amount of shadow can conceal. This is the world that certain snakes inhabit. Pit vipers, pythons, and boas possess sensory organs that have no direct equivalent in any other group of vertebrates. These are the pit organs, small membrane-lined depressions positioned on the face, between the eye and the nostril, or along the jaw. Each pit contains a thin membrane stretched across an air-filled chamber, and this membrane is extraordinarily sensitive to infrared radiation, the heat emitted by any object warmer than its surroundings. When a warm-blooded animal moves within range, the snake detects the difference in infrared emission between the animal's body and the cooler environment around it. The result is something analogous to an image, not a visual image formed by reflected light, but a thermal map, a representation of the world in gradients of warmth. The resolution of this sense is remarkable. Pit vipers can detect temperature differences of as little as a fraction of a degree Celsius. They can locate and strike prey with high accuracy in conditions of total darkness. The pit organs work in concert with the snake's conventional eyes, providing a layered, multimodal picture of the environment that is richer than either sense alone could produce. We see a world made of light. They see a world made of warmth. Both are real. Both are the same world, perceived through entirely different windows. For most of the year, the Arctic tundra is a landscape of extraordinary restraint. Snow covers the ground from September until late spring. Temperatures drop to levels that would be life-threatening to most organisms without significant physiological preparation. The wind moves across the open land without obstruction, carrying cold from the interior of the continent and the polar sea. And then the sun returns. In the high latitudes of the Arctic, summer is not a gradual warming, but an event. The sun climbs higher each day until near the solstice it barely sets at all, tracing a low arc across the sky through the night, never quite disappearing below the horizon. The permafrost surface thaws. Standing water appears in low depressions. And the land, which appeared entirely dormant just weeks before, transforms. The bloom happens fast. Flowering plants that have waited through the darkness complete their entire reproductive cycle in a matter of weeks, producing blooms of purple and yellow and white across the tundra surface. Mosses green almost overnight. Insects emerge in numbers that seem almost impossible for a landscape that was so recently frozen. Migratory birds arrive in vast numbers from wintering grounds thousands of miles to the south, drawn by the explosion of insect life and the long days that allow almost continuous foraging. Caribou calve on the open tundra. Arctic foxes, already in their summer coats, raise their young. All of it happens in a window of perhaps 8 weeks. Then the light recedes, the temperatures fall, and the tundra draws itself back into stillness. It is one of the most condensed, urgent expressions of life on Earth, a season that burns bright precisely because it is so brief. On the seafloor, moving with the unhurried patience of something that has never needed to hurry, the sea cucumber goes about its existence. It is soft-bodied, roughly cylindrical, and to a casual observer, it does not appear to be doing very much. It processes sediment. It breathes through an unusual arrangement involving its own digestive tract. It moves, when it moves at all, with a slowness that makes other slow animals seem rapid by comparison. But inside that apparently simple exterior, something remarkable is possible. A sea cucumber can change the mechanical properties of its own body tissue on demand. When threatened, or when it needs to squeeze through a narrow crevice in the reef, it can alter the arrangement of collagen fibers in its body wall in a matter of seconds, transitioning from a firm, structured state to something approaching liquid. The body softens and flows, conforming to whatever space is available, then firms again once the animal is through or the threat has passed. This ability, mutable collagenous tissue as biologists call it, is controlled by the nervous system through chemical signals, and it operates independently in different regions of the body. The sea cucumber can be firm at one end and soft at the other simultaneously. Some species extend this principle of defensive self-modification further still, expelling portions of their own internal organs when threatened, leaving them behind as a distraction for predators while the animal itself escapes. The organs regenerate over the following weeks. The sea cucumber dissolves and reforms itself as quietly as breathing. In the slow world of the ocean floor, it is, in its own way, extraordinary. In the waters surrounding Antarctica, where the sea temperature hovers just below the freezing point of ordinary fish blood for much of the year, a group of fish has evolved a solution to a problem that should, by all normal biological logic, be fatal. The icefish, 16 species belonging to the family Channictidae, carry antifreeze proteins in their blood and body fluids. These proteins bind to the surfaces of any ice crystals that begin to form within the fish's tissues, preventing those crystals from growing into structures large enough to damage cells. The antifreeze proteins do not lower the freezing point of the blood substantially. Rather, they interrupt the process of ice formation itself, holding it in a kind of suspended state. The icefish have made other adaptations to life in Antarctic waters that are, in their own way, equally striking. They have no hemoglobin, the iron-containing protein that gives the blood of most vertebrates its red color and its capacity to carry oxygen efficiently. Icefish blood is pale and thin, almost translucent. Oxygen is carried dissolved directly in the plasma, a far less efficient method that the fish compensate for with unusually large hearts, wide blood vessels, and high blood flow rates. They move slowly through water that is perpetually close to freezing, in an environment where the seafloor is largely barren and the light for half the year does not reach. Their existence is quiet, cold, and spare. But they are alive in a place where being alive requires a biochemistry that evolution arrived at nowhere else on Earth. Rising from the red soils of tropical Africa and Australia, termite mounds can reach heights of 9 meters or more, towering structures of cemented earth that are, relative to the size of the creatures that built them, among the largest constructions produced by any animal on the planet, including humans. But the most remarkable thing about a termite mound is not its height. It is what happens inside. The interior of a large termite mound is a complex architecture of interconnected chambers and passages. Near the base, broad air tunnels run close to the cooler soil. Higher up, narrower passages branch outward toward the mound's surface. The mound's outer walls are thick in some places and thinner in others, in a pattern that is not random. The result of this architecture is a passive ventilation system of considerable sophistication. As the sun warms the mound's surface during the day, air inside expands and rises, drawn upward and outward through the network of passages. Cooler air from the base tunnels flows in to replace it. The gases produced by the colony's respiration, carbon dioxide and moisture, are exchanged for fresh air through this continuous convective cycle. The internal temperature and atmosphere of the mound remain remarkably stable, maintained within the narrow range required by the colony's fungal gardens, which the termites cultivate as their primary food source. No blueprint was drawn. No engineer was consulted. Each termite followed simple behavioral rules determined by its caste and the local chemical environment. And from the collective behavior of millions of individuals, following those simple rules, an air-conditioned city emerged. In the arid scrublands of western North America, a small flattened lizard moves slowly across the ground, armored with pointed scales and a rounded body that gives it, in certain lights, the appearance of a small horned toad. It is called the horned lizard, and it is a gentle, unhurried animal that feeds mostly on harvester ants and spends much of its time basking in the sun. But if a coyote, wolf, or domestic dog investigates too closely, the lizard deploys one of the most startling defensive mechanisms in the vertebrate world. It shoots blood from its eyes. The mechanism involves the sinuses around the lizard's eyes, which can be engorged with blood by the restriction of outgoing blood vessels, causing pressure to build until small blood vessels in the eyelid rupture. A thin, pressurized stream of blood, mixed with chemical compounds derived from the lizard's ant-heavy diet that are particularly repellent to canine predators, is ejected from the eye socket, sometimes traveling several feet. The response is effective against dogs, coyotes, and wolves, which appear to find the compounds in the blood highly aversive. It is less effective against raptors and other non-canine predators, which the lizard addresses through other means, primarily its camouflage and stillness. The lizard is not harmed by the process. The vessels heal quickly. The blood supply is replenished, and the lizard returns to its slow, careful life in the scrub, no worse for its defense and rather less troubled by whatever startled it. Before the first fish, before the first shell, before the first cell with a nucleus, before almost everything we associate with the complexity and diversity of life on Earth, there was the deep ocean. At the bottom of Earth's ancient seas, in places where the pressure was immense and the darkness total, and the temperature swung violently between the freezing cold of the surrounding water and the superheated mineral-rich fluid venting from fissures in the ocean floor, something began. Hydrothermal vents, cracks in the seafloor through which geothermally heated water escapes, carrying dissolved minerals from the rock below, create a chemistry at their margins that some researchers believe may have been the cradle of life on Earth. The mixing of hot, mineral-rich vent fluid with cold seawater produces gradients of temperature, pH, and chemical concentration that could have driven the earliest biochemical reactions, the first tentative steps toward metabolism and self-replication. The earliest organisms that took hold in these environments did not photosynthesize. They could not, there was no light. Instead, they drew energy from the chemical reactions between the minerals in the vent fluid and the surrounding seawater, a process called chemosynthesis that still sustains the communities living around hydrothermal vents today. Today's vent communities, tube worms, clams, shrimp, and heat-tolerant microbes living in water that would scald any ordinary organism, are a window into something very old. In the dark, at the bottom of the sea, where the heat of the earth meets the cold of the ocean, life may have written its first sentences. The starfish moves across the seafloor with a patience that seems almost deliberate, 5 arms spreading outward from a central disc, each lined beneath with hundreds of tiny tube feet that grip and release the substrate in coordinated waves, pulling the animal forward at a pace that makes the word slow seem almost inadequate. If a predator seizes one of those arms and tears it away, the starfish does not bleed out. It does not succumb to infection. It seals the wound and then, over the course of weeks or months, depending on the species and the conditions, it grows the arm back. Not a simplified version. Not scar tissue filling a gap. A complete, fully functional arm, with all its tube feet and sensory structures intact. Some species of starfish can do something even more remarkable. If the severed arm retains a sufficient portion of the central disc, it can regenerate not just the lost arm, but an entirely new body, growing a complete individual from what was, moments before, simply a fragment. This capacity depends on a distributed population of undifferentiated cells, capable of transforming into whatever tissue type the regenerating body requires. An approach to healing that is fundamentally different from the scar-based repair used by most vertebrates, including humans. Scientists studying starfish regeneration are working to understand the molecular signals that allow this process to operate with such fidelity and completeness. The hope is that understanding how a starfish rebuilds itself might eventually offer insight into how human tissues might be persuaded to do something similar. In the quiet of the reef, the starfish grows on. When the first preserved specimen of a platypus was sent from Australia to Britain in 1799, the scientist who received it examined it carefully and then checked the body for signs of a taxidermist's stitching. He was convinced someone had sewn a duck's bill onto the body of a beaver-like mammal as a hoax. He found no stitching. The platypus was real. It remains, more than 2 centuries later, one of the most biologically improbable animals on Earth. The platypus is a mammal, warm-blooded, furred, and capable of producing milk to nourish its young. But it lays eggs, as reptiles do. It has no nipples. Milk seeps instead through pores in the mother's skin, and the young lap it from her fur. The male carries a venomous spur on each hind leg, capable of delivering a toxin that causes severe pain in humans and is lethal to smaller animals. The composition of the venom is unique, containing compounds found in no other venomous animal. When hunting underwater, the platypus swims with its eyes, ears, and nostrils closed. It navigates and locates prey entirely through electroreception, the detection of the tiny electrical fields generated by the muscle contractions of other animals. Its bill contains tens of thousands of electroreceptors, making it one of the most sensitive electrosensory organs known in any vertebrate. The platypus represents a lineage that diverged from other mammals more than 160 million years ago. It has had that time to become exactly what it is, a creature that follows its own rules, laid down long before ours existed. As daylight fades and the insects begin to rise from the vegetation, bats emerge from their roosts, from caves and tree hollows and the spaces beneath roof tiles, and take to the air. Within seconds they are hunting. They cannot rely on vision alone in the dark. Most of the species that hunt on the wing have evolved something far more precise. Echolocation works like this. The bat produces a pulse of sound, typically at frequencies well above the range of human hearing, between 20 and 200 kHz, through its mouth or nose. This pulse travels outward into the environment, reflects off any object in its path, and returns to the bat's ears as an echo. By analyzing the properties of that echo, its timing, its frequency, the way those frequencies have shifted through the Doppler effect, the bat constructs a detailed representation of the space around it. The resolution of this system is extraordinary. A bat using echolocation can detect a wire 2 millimeters in diameter in total darkness. Some species can distinguish between moth wings of slightly different thicknesses. The time between emitting a pulse and receiving its echo gives the bat the distance to the object. The way the echo's frequency has shifted tells it whether the object is moving and how fast. Some bats produce pulses at rates exceeding 200 per second when closing in on prey, creating what is essentially a continuous acoustic image of the world around them. They do not see darkness the way we do. They hear space, and in that heard space, the night is full of shape and movement and information. In the low, waterlogged soils of the coastal plains of North and South Carolina, where the ground is perpetually damp and the sunlight is generous, but the soil is poor in almost every nutrient a plant requires, the Venus flytrap has arrived at a solution that took evolution millions of years to refine. It waits. And it counts. Each of the flytrap's jaw-like leaves is lined on its inner surface with a small number of trigger hairs, typically 3 on each lobe. These hairs are exquisitely sensitive to mechanical contact, but the trap does not close on the first touch. A single hair bending in the breeze or a raindrop landing on the leaf is not enough. The plant requires confirmation. If a single trigger hair is touched twice, or if 2 different hairs are touched within approximately 20 seconds of each other, the trap closes, snapping shut in a fraction of a second, one of the fastest movements in the plant kingdom. This 2-touch requirement serves as a filter, distinguishing the irregular, repeated contact of a moving insect from the single chance contact of a falling leaf or a drop of rain. Once closed, the trap seals progressively tighter as the insect continues to struggle. Additional trigger hair contacts signal that the prey is alive and worth digesting. The plant secretes enzymes,